Okay, so can everybody kind of see this? Looks like I've got all my technology working here. Everybody good? Uh, okay, so how's everybody doing?
Good.
Excellent. Yeah, hopefully you can kind of see the screen. If you can't, like I said, I'll at least, I can put up a PDF of the slides. And, maybe there will be a screen recording that works. We'll see how that goes. So I'll talk to you a little bit today about fMRI and EEG and various other techniques for studying the brain, mostly the human brain, although we'll talk a little bit about other stuff. I guess you guys are only on day 2 of this, so I don't know exactly what you know, and it sounds like you guys are coming from different places and might have studied different stuff before this, so feel free to stop me at any time. I've got various comic strips and things peppered throughout to illustrate some points. One of the themes that we'll be talking about, if you can't read this, you can read it later, but basically the joke in this comic is that, the guy is, this always makes things funnier to explain them in detail with science. But the idea is that this guy is studying human behavior by talking to a woman at a bar, which is sort of the old school. That's not how we used to study the human mind per se at bars. Although there was a functioning bar in the basement of one of the Yale psychology buildings a few years ago when they were doing alcohol studies. So they built like a whole fake bar for people to go in and drink in. But usually that's not how we do it. But you did have to kind of just give people stimuli, right? Some sort of sensory experience and then record what their behavior in some way. You couldn't get inside their actual brains, right, until really a few years ago most of the time. So now we can do things, and this is what the second comic kind of alludes to, we can record from the brain, and the dream is to one day be able to understand the brain well enough that we can kind of predict behavior and kind of understand how the brain gives rise to behavior and so forth, right? So that's all that stuff. So just to remind you, so this is me. I'm Matt Johnson. Here's my email address. If you ever have questions after this, feel free to ask questions throughout the talk. But if you have questions later on, you can always email me or whatever. Okay, so just a little bit of background on me and how I came to be here in this room. So I did my undergrad back at Yale a long time ago. Then after that, I worked for a little neuroscience research institute in Hartford, Connecticut, where I learned to do functional MRI mostly, and we did various types of studies with different psychiatric disorders and so forth. Did that for a couple years, then went back to Yale to do my PhD, where I met such fine individuals as a person then known. Oh wait, well sorry, that's not you. I anticipate. Sorry, I worked with my advisor, Marcia Johnson. No personal relation, no family relation to myself. But she is my academic mother now. So this is her playing the Wii. So all of these studies that I'm going to talk about are done in collaboration with Marcia and various other people in Marcia's lab. This is the person formerly known as Sarah Gray, now Dr. Sarah Richards, and her husband I think this is Halloween 2008. This is one of the innocent pictures. So Sarah, a science lover as always, was dressed as a cannulated rat, I believe. For just full disclosure, that was the year I went as presidential or vice presidential candidate Sarah Palin for Halloween, so I didn't want to embarrass you without also doing one on me, but you notice I didn't leave that up very long. And then after finishing, so Sarah and I finished our PhDs around the same time. After that, I took a job in a somewhat random move as an assistant professor of psychology at the University of Nottingham, Malaysia campus. So this is Malaysia, where I lived for a couple of years, taught psychology and did research there. Then had enough of that and decided to come back to the US, and actually I'm now back at Yale one more time, back with Marcia Johnson, doing a postdoctoral role in her lab for a little while, doing research there. And I'll probably be doing that a little bit longer, and then hopefully go back on the job market and get me a real job. Okay, so that's sort of who I am, what I do. So I do a lot of different types of cognitive neuroscience and neuroimaging and stuff, and we'll talk about, I think, all of that today. Before we start, this is, again, just to remind you, this is something I will not be doing today. I will not be breaking out the condescending cat slide if you guys ask a quote-unquote stupid question today, right? So, like, anything's fair game. Feel free to ask me whatever you want. I reserve, if it's too personal, I reserve the right not to answer. But But yeah, if like something doesn't make sense, if I didn't give you context to understand something, or just I'm going too fast or whatever, I'm a little caffeinated today, as I am most days. So like I will go quickly, just because that's the speed that my brain operates at right now, especially on a lot of coffee. So like slow me down if you need to, or just stop and ask questions, or if you want. If you would like me to go off on a tangent, I'm really great at that. So like feel free to send me off on if you have questions. And just like since it's dark, just like attract my attention however you can if I don't see your hand or whatever. Okay, so I figured I'd start out talking to you guys a little bit about a bunch of different neuroimaging methods. I mostly use fMRI and EEG and do some just pure behavior stuff. But you know, in this field, you kind of run across a lot of different methods, and you have friends that do different types of methods, and so on. So I thought I'd kind of review the various ways that we have of investigating the brain at different spatial scales and different time scales, and some of the advantages and disadvantages of all of them, just to kind of get you, give you an idea of like what we can and can't do in neuroscience research, because there's a lot that we wish we could do better, and you know, a lot we can do pretty well. So if we really wanted to, you know, if you wanted to be able to perfectly predict what someone was going to do, like a futuristic fortune teller, I mean, really what you would need is a perfect recording of their brain activity, right? Because you've got, I think the best estimate I've seen so far, you've got about 86 billion neurons in your head, and each of those has the capacity to fire an action potential, to like, you know, to fire a little bit of activity up to several hundred times a second. So, you know, that's a lot of information that's zipping through your brain at any given moment, right? So, you know, if we really wanted to have a complete uncompressed version of what the brain is doing, we would need a lot of data, right? We need a recording method that somehow could measure all of those brain cells simultaneously, could do so with a high sampling rate, meaning, you know, that we can, you know, like your video runs at 30 frames a second or whatever. So we want a certain number of frames a second of data, right? So ideally, we would be able to get data at a rate of over 1,000 hertz, meaning, you know, under 1 millisecond sampling rate, because it takes about 1 millisecond for a cell to fire an action potential. So ideally, we'd be able to record a little bit faster than that to get the full picture of what the brain's doing. It would be great if it did not cost very much. That's always a nice thing, because we can then buy more cars and you know, nice food and stuff for the researchers if we don't have to spend all of our money on research. And it would be nice if this did not inconvenience the participant, right? By having whatever nanobots injected into their brains or something like that and taking over. So that would be nice. And these constraints or these factors we kind of refer to with various names. So spatial resolution is what we use to refer to like how, just like your resolution on your phone screen, right? Like, you know, how in space, how precise your measurement is. In time, we call that temporal resolution, so how fine-grained you're able to get in time with your recording. So again, that's sort of like your, you know, your 30 frames a second or whatever of a movie. We'd like to get more like 1,000 frames a second if we could. Cost is just cost. That's no big deal. And we usually refer to, when we talk about convenience, we're really talking about invasiveness, like how much do we have to, you know, trouble this person or like invade their brain to record from it. So we would ideally like to have great spatial resolution, great temporal resolution, low cost, and low invasiveness. And of course, that's not going to happen ever. That's impossible. That's like having a car that, you know, goes 1,000,000 miles an hour and you know, gets 1,000,000 miles to the gallon, right? It's not physically possible. So we make the compromises. We have to try to, you know, try to use different methods and try to infer what we can from what we know in different disciplines, and try to get a general sense of the big picture, even though we can't get everything all at once. Okay, so just to kind of give you an orientation of like spatial scale here, kind of the sort of the smallest thing that a neuroscientist like me would ever think about doing is a single cell recording. That's where you, well, we'll talk about more, but that's where you're recording from a single neural cell. So that's a few microns big. So that's, you know, very, very small spatial scale. Next thing up is a pretty big leap in spatial scale. So that's functional MRI, which usually we're looking at chunks of brain about 2 or 3 millimeters wide by 2 or 3 millimeters by 2 or 3 millimeters. PET and SPECT we'll also talk about. So that's a little bit bigger than fMRI, like 4 or 5 millimeters usually. There's something called intracranial EEG. That's more like we get 1 centimeter or so is about how widely spaced those sensors are. Then there's something called MEG, and again, we'll talk about what all these actually are. They're just names right now, I know. That can be anywhere from several millimeters to a few centimeters. So that's kind of spans this whole range in various ways. Then there's regular EEG, where we're really talking about kind of several centimeters apart is where we're sensing things. And then if you really want to go beyond what I would call cognitive style neuroscience, you've got stuff more like what I think Sarah did, down into sort of molecular biology and biochemistry, where you're working really with individual cells or molecules or whatever. If you want to really go, we're all just particles flying around the universe. If you want to really break it down and treat us all as quarks and electrons and so forth. But of course, that's not a super useful way to characterize something like a human brain. But you can go all the way down if you want to. And then on the other side, of course, like you've got things that don't really explicitly involve the brain at all, but cognitive psychology, obviously, whether or not you study with neuroscience methods or not, you are studying the mind, which, spoiler alert, is created by the brain, right? Like that's how we have minds, is you have brains. And then, you can go on from there to like groups of people, social psychology, and then really big groups of people like economics and political science and so forth. Right, so again, I wouldn't really call these neuroscience methods, but they're kind of you kind of keep expanding the spatial scale out to infinity. That's what you get. Okay. So we also talked about speed and sort of what our temporal resolution is, how fast we can record. So just to give you an idea of how all these things work, again, we'll talk about these in more detail. Single cell recordings are very fast. You can get them kind of as fast as you can make your computer record data, so under 1 millisecond. 10 samples a second, I think, is not uncommon or more. Intracranial EEG and MEG and regular EEG are all pretty good. You can get about a sample every millisecond here. And also these you can kind of go arbitrarily fast if you want to. It's just you end up with a lot of data if you go too fast. Go through a lot of hard drives that way. Functional MRI is significantly slower. So you can record functional MRI images every like 1 to 3 seconds. But as we'll talk about a little bit later, the story is actually even worse than that, in fact, with functional MRI. It's not the fMRI's fault, it's your fault. It's your stupid brain and your stupid blood's fault. We'll talk about why that is in a minute. Not just yours, mine too. I don't have special blood. Then there's PET and SPECT, which is even worse. It takes several seconds to even minutes to get a PET or SPECT image. And that's it. And the other stuff doesn't really make sense to talk about temporal resolution. So a lot of different timescales we can record on also. And again, none of these methods is perfect, which is why we have all of them. Then there's a couple other things we can talk about that aren't really quote-unquote recording methods, but you can use to study the brain directly or indirectly. There's things called transcranial magnetic stimulation or transcranial direct current stimulation, where we actually try to affect the brain. And you can also do lesion studies, which I think Sarah kind of talked about a little bit this morning, like if something goes wrong with your brain, what happens, right? So, we'll talk about that a little bit also. Okay, any questions like so far? That's just kind of an overview of all the junk that we could talk about today. Anybody got questions so far? This will get a little bit more interactive and a little bit not. I didn't make it super interactive because I wasn't sure how many of you guys there are, but there will be some interaction. But feel free to, so no questions so far? Good. Okay. So just to talk about a little bit about the stuff that I don't know that much about myself, because I don't do it. So single cell recordings are pretty simple, pretty easy idea in theory, right? So I don't know how much you guys know about neural cells so far, but you probably know that they communicate biochemically with like neurotransmitters, but one thing those neurotransmitters do is affect the voltages of the cells in relation to their surrounding environment. So although they're not electrical the same way that like a computer's electrical, there are voltage differences involved, and changing the voltage in the brain or kind of around a cell affects its function. So the idea of and conversely, when a cell, for example, fires, that creates a voltage change that you can measure. So single cell recordings are pretty simple in principle. You take a very thin wire electrode and stick it into the brain and record whatever voltages it picks up. So record when a cell nearby fires. It creates a little blip in your voltage recording, and you can, record that on a computer. Back in the old days, I guess you would do that with an oscilloscope or something like that. And then you analyze your data and get your Nobel Prize or whatever. Or like some of our friends, you languish in grad school for 8 years, like cursing the whole thing because it's actually really hard to do this stuff. But one of those two things will happen. So that's the basic idea. You just stick an electrode down in the brain and record the electrical activity going on nearby. And so usually you would not do this with a human being. Usually you would do it with a rat. These are both very, clearly technically correct diagrams of rat electrode. It's just basically showing you've got a wire in the animal's head, and that's connected up to a computer that does the recording, essentially. And meanwhile, the rat is, probably doing some sort of task. Maybe looks like it's got some lights to look at or something. The monkey is busy writing his novel or doing something. No, he's probably like, doing some kind of computer-based task, while we record from their brain cells. So there are some advantages of doing this. It's got great temporal resolution, like we said. You can get really fast recordings and look at individual firings of individual cells, which is pretty cool. And it's got really good spatial resolution, so down to the microns level. So you can typically record from 1 to several neurons, sort of depending how you do this, and you can make little arrays of multiple electrodes to get multiple neurons simultaneously, but still not very many. So the disadvantages, there's a lot of those too. You can really only record from a few cells at a time. Now again, if you have one of these big arrays, you might be able to get a couple hundred or something, but that's a couple hundred out of billions, right? And in a very restricted region, right? And obviously this is extremely invasive. You can't just poke a wire down into your head. You kind of have to make a hole in your skull first, right? So, and this is something that I think upsets animal rights people, and somewhat understandably, I think if you don't know too much about this, I won't go on the whole rant. We have some of our best friends who are wonderful and empathetic and kind people do animal work, and I'll just say that the animals that have this done to them live pretty good lives for animals. They you do have to give them surgery to make a little temporary hole in their skull so that you can poke the electrode down in their brain. But they get anesthesia just like a human brain surgery patient does, and they get Tylenol and stuff afterwards just like a human would. So they're allowed to recover and so forth. And they don't. Most of them, the monkeys can be jerks a lot of the time, as can the rats. But for the most part, they don't seem to mind it too much. They're pretty happy-go-lucky as monkeys go.
How can monkeys be jerks?
In so many different. We have a friend who might have gotten a monkey bite, although she might have taunted the monkey a little bit too much, I think. Monkeys, just like humans, they'll refuse to work sometimes. They have been known to throw bodily outputs in various ways, they do all kinds, or they can just be uncooperative. Do you have a question?
Yeah, so with this method, would you have to know which region of the brain you wanted to measure before you used it? And I guess what kind of situations would this method use?
Yeah, that's a good question. So yes, you would definitely, if you've got billions and billions of neurons, I guess a monkey doesn't have as many as a human, and a rat has even less, but you've still got billions of neurons to worry about. So yeah, you usually have a very specific hypothesis that you're testing. So one example, so we have a good friend who you'll actually see an appearance by right at the end of this talk, named Matt Krause. So a friend of ours does monkey single-cell recordings, and he's interested in vision. So they record from different areas, but usually you have a hypothesis about how a certain visual area works. And this kind of actually is a good point to mention that there's synergy between these different methods. So a lot of this, not entirely, but a lot of the same brain areas or analogous brain areas are present in humans versus monkeys. And it's a little bit less exact, but it's there's also correspondence between rat brain and human brain and so forth. So you might find an area or investigate an area in a human neuroimaging study and say, oh well, I found this area that appears to do this. But I don't have a certain level of detail about how that information is actually processed. So now that I know this area, exists in humans and I know where to find it and so forth, now I can go plunk some electrodes in a monkey's brain and kind of look with finer detail at that area. So actually, funnily enough, this is a slide I actually took out of this exact point in the talk that basically talks about exactly this for time purposes. But for example, in humans, we have a brain area that's called the fusiform face area. You guys, I don't know if you've heard of it. It's very famous, probably a little too famous because it's a little more complicated what it does than just analyze faces. But this brain area activates a lot when you see a face, okay? So, and it doesn't activate very much for most other things. So, we don't still know exactly how it does it or what it does, but we know it somehow is involved in visual face processing. So we know that this area exists in humans. And one thing we also know is that if you put an electrode in a monkey's brain at kind of a corresponding location and show it faces and other things, you can actually see, find individual cells that fire a lot when you show a face and fire not so much when you show something that's not a face. And again, I took this slide out. I had a perfect illustration of this, but basically, yeah, I'll get you in a second. So they did a study, for example, where then they could find a cell that liked faces, and then they'd show it a face, and they'd show it a face tilted a little bit to the side, and they'd show it a face like a human face or a monkey face, or they'd show it like a toilet, like one of those fuzzy kind of toilet brush pictures that kind of has the outline of a face and is sort of fuzzy like a monkey, but isn't an actual face. And you can do things like measure the cell's response to all these different types of things, and get a much finer grained amount of information as to what that cell does versus this human brain area that's got lots and lots of cells in it, and you're kind of seeing the average of all of them all at once. So yeah, that's a roundabout way of saying that yes, you would always have a very specific hypothesis, and you'd have some idea about what area and what cognitive process you're interested in, and then you kind of plunk your electrodes down and go hunting for cells that participate in that process. Yeah.
So if you narrow down the specific region that you want to study using this method, how exactly do you pinpoint where you're going to put each electrode? If you have more than one electrode.
Yeah, it's tricky. There's various ways you can do it. I mean, brain anatomy is somewhat. Are you guys going to do? You guys did some neuroanatomy already, right? So did you find the central sulcus and stuff like that?
Sure did.
All right, excellent. So as you guys have found but you haven't worked with a lot of actual brains, right?
We dissected a sheep brain.
Oh, a sheep brain. Okay, cool. Awesome. So you guys have seen sheep brains. So human and sheep and other brains, individuals have a lot of the same kind of basic organization of the brain, right? We all have a frontal cortex and we all have an occipital lobe and so forth. And we all pretty much have a central sulcus and we all pretty much have some of the major landmarks of the brain. But the fine details, aside from sort of the major landmarks of the brain, then it gets kind of complicated. Brains are pretty different from one another. Fortunately, less so in monkeys. So human brains, and aside from just being larger than monkeys, you guys know, well, you've seen how the brain surface has all these convolutions, right, and is all folded in on itself. So the monkey's brain surface is a little bit smoother. So monkeys tend to have a little bit less variability in their brain structure, but still there's variation between individuals. So to some extent, it's a little bit of approximation. But one thing you can do is, so what you typically do with a monkey is you have what's called a stereotaxic apparatus. So basically, you put the animal's head kind of in this. Has anybody seen Back to the Future?
Yes.
Okay, you know that initial scene where Doc Brown has that weird, looks like kind of a bunch of girders on his head that he's trying to use to read Marty's brain. It's like a head cage kind of thing. So it fits on his head. So it looks sort of like that. It's this kind of head cage thing, and the idea is you put it on the head, and then you've previously mapped out in other animals kind of what head position corresponds to what brain position. So you can sort of find a point on the surface and go down from there to find a standardized coordinate for where this brain area is supposed to be. So that's one way you can do it, is with this kind of stereotaxic guess by just kind of putting the head in the standard position and using this kind of framework apparatus to pinpoint the coordinate where it's been found before. The other thing you can do, and this is a little bit trickier, is you can scan the monkey's brain using MRI. You might even try to get the monkey to do functional MRI, although that's a lot harder. So you can either make the monkey go to sleep, not just like bedtime, but like, give it a medication. What's the word I'm looking for? An anesthetic to like knock it out for a couple of hours and scan its brain just to get the monkey's brain structure. And then you can actually like get a picture of what that monkey's brain surface looks like and get a much more detailed guess as to where their functional areas are going to be. Or some people that are really patient have been able to train monkeys to lie still in an MRI machine and do actual tasks like a human would in an MRI machine. And then you can actually find that particular monkey's functional areas and see what parts of the brain are kind of lighting up. And we'll see like these examples later on, like these little blobs that you often see in the news if there's a neuroscience study. So you can actually get that monkey's specific functional areas and then, translate that to outside the scanner and kind of, find on the surface of the head where that area would correspond to and then poke your electrode right in there. Yeah. So there's a few different ways you can do it with varying degrees of effort and guesswork involved. Any other questions? Okay. So that's single cell stuff. We won't talk much more about that. Sorry. Come on, buddy. Okay. So yeah, this is usually because you've got to like, cut somebody's head open, make a little hole in their skull, and kind of, keep that hole open long enough so these recordings it usually takes like a whole day to just record from like a few cells. So usually you spend a day or several hours in a day, kind of like you trot the monkey out. I guess less so with rats and stuff. You can probably go a little bit quicker with rats. Maybe not. I've not had to do it, but. You spend a few hours in a day kind of getting the monkey out of the cage, holding his hand, walking him down the hall, like getting him set up in his little chair. And then he does the task for a couple hours while you try to find, a good cell to record from and try to record from that one cell for a couple of hours. And then you kind of put the monkey away and like, back up the data and so forth, and do that all again. So if you do a study where you study like a couple of hundred cells, that might take you half a year to do. I have not personally, but friends of ours have done this and have like labored with this.
The same guy who's going to apparently come up at the end of the talk, Matt Krause, he did recordings in conjunction with another guy who was in grad school with us from cats. And their cat recordings would be up for, I think it was between 24 and 36 hours straight.
Yeah, they would do those in the middle of the night. So the cats, it's a little bit different. We won't get into the full details, but the cats were like anesthetized and stuff, and it was because the cat. So you had to like make the cats unconscious for this because this wasn't studying active behavior. This is studying sort of passive visual processing, and because of that, like you've got to keep the cats like on a respirator, and also like it's real intensive stuff. It's not like an awake behaving monkey. So yeah, you want to kind of record from the cat as long as you can. So yeah, you're basically staying up all night to do that. They were up all night to get data.
Many nights.
Yeah, many nights. Yeah, it was like a whole ordeal for them, and it often didn't go very well if they didn't get good cells or whatever. So it takes real dedication to do this kind of work. But yeah, anyway, so because of all this, this is not something that you would do to a human, right? You wouldn't. There are people that would think that this is not something that you should even do to animals. I personally disagree. I think we get really useful information for helping the world out of, doing these animal studies. But, you usually would not crack a human's head open and like implant an electrode permanently in their head to like, just learn how vision works or whatever. Although I would be, like I volunteer for studies all the time. You can open up my head if you want to, but usually our ethical review boards do not let us do this. However, there are occasions, so a lot of interesting data has come out of people with epilepsy. We'll talk about this a little bit more later. So people with really severe epilepsy often have, and some other disorders as well, will sometimes have electrodes implanted in their head, usually to kind of study where the seizures are coming from, so that if they have to have surgery, you can take out the areas that are causing the seizures, or sometimes to deliver bursts of stimulation in certain disorders to alleviate the symptoms. So anyway, so some people have electrodes implanted in their brains for medical purposes anyway, and then so every once in a while we get to make single cell brain recordings from human beings, but usually we don't get to do that.
Quick addition to that, one of is they were looking and they saw that within the fusiform face area there was a cell or an ensemble of cells that fired specifically to pictures of Jennifer Aniston.
That is
and
correct.
they call it the Jennifer Aniston cell reporting paper. It's a pretty famous
Yeah.
paper, and they can show her any picture of Jennifer Aniston, whether she was
Yeah, and you could show the name Jennifer Aniston.
Yeah.
Now, this was only in one person. They happened to find one cell that worked like this. Yeah, but, if you showed the name Jennifer Aniston, it lit up. I think if you showed Jennifer Aniston next to Brad Pitt, though, it would not fire. Like,
It would, like,
Brad
inhibit
Pitt inhibited
it,
the
I
Jennifer
think.
Aniston
It was
cell, yeah.
very funny.
So actually, this forecast to the end, I was going to put in a quick plug for my podcast with my buddy Matt Krause, who we just were talking about. So we actually talked about that exact study on the episode that we just recorded, and which I will be editing and releasing hopefully soon. But we actually talked about that, and maybe a teaser for both the podcast and the research. That same research group just recently published another single-cell human recording study where they may have found the seat of human consciousness. It's probably not that exciting, but this group is very interested in consciousness, and there's a brain area they think has a lot to do with consciousness, and they did a single-cell recording in a lady that was pretty interesting. Okay. So any other questions about the single-cell stuff before we go on? Cool. Okay. Oh, the clicker guy is not doing his job today. Okay, so now we'll move up. So I'm going to mostly kind of zip through the stuff that I don't personally do early on and leave the real heavy stuff that I focus on for later. So there's also something that we call, so this is more of a real human neuroimaging method, although you can also do it in monkeys and so forth. There's two actually called PET and SPECT that are in many ways very similar, so I kind of lump them together. A lot of people lump them together. These are short for positron emission tomography and single photon emission computed tomography. Those are long names for basically saying that what you do is you inject someone with a small amount of a radioactive substance. That substance will cause their blood or whatever the substance is to emit either positrons or photons, hence the names. And then you can compute tomography, which is just kind of a brain map or a map in general of where those cells are coming from that radioactive substance. So the idea is you inject them with the radioactive substance. Okay, I'm just going to give up on the clicker. Put the person in a PET or SPECT scanner, depending on what type of study you're doing, and see where the radiation ends up. Okay, so needle, scanner, done. That's how it goes, right? Sorry, that's like super bright. They aren't really, it's not like the gates of heaven normally. Like, it's not only that bright, it's a really bright display. Yeah, so the basic idea is, you can tag all types of chemicals with a little bit of radioactivity. So you can take, so for example, as we'll see on the next slide, you could take glucose, for example, and I forget exactly what it is, like a little bit of radioactive something. I forget which element it actually is, and kind of hook it onto the glucose molecule and see where the glucose goes in the brain or the body. So like I said, these are similar conceptually. The only difference is PET uses radioactive materials that emit positrons when they radiate. SPECT uses photons, and then you have kind of the person lies in a scanner that's kind of a particle detector, and you can compute where these particles are coming from, basically. It was the main way that we did cognitive neuroscience until fMRI, which the sort of very first indication that we might be able to do fMRI was around 1990. By about 1995, people were actually getting it working pretty well for real studies. So these, like I was alluding to just a second ago, these PET studies usually used what was called radiolabeled glucose. So you take, one atom in the glucose molecule, and I'm no chemist, so I don't know exactly how they do this, but you replace it with a radioactive atom of some sort. But it basically still functions as glucose, and then you inject that in the person's blood. When you use your brain uses glucose, so the areas of the brain that are active take up more of the glucose. Because that glucose is radioactive, it shoots out little positrons, which the detector detects, and you know which areas of the brain took up the glucose, and thus which areas of the brain were active, right? So basically, those PET images tracked energy metabolism in the brain. Now, there's other ways you can do, so the kind of cool thing about PET and SPECT is you can tag, and when I say tag, I just mean hook a little radioactive atom to. So you can tag a lot of different types of molecules. So other PET studies just use radio-labeled water. So you just have, I think it was deuterium, I guess, right? I think that's what it is. Was with radioactive hydrogen in the water instead of just regular water, which is hopefully not radioactive most of the time. So that would do the same thing, except it would just track like actual blood flow, right? Because you inject radioactive water into your bloodstream, and then that just follows the blood around. So that's another way you could do it. And you can also use it for kind of cool stuff. To skip ahead a point, like look at things besides just general activity. So if you're tracking where like blood flow goes in the brain or where glucose goes, that's just kind of tracking like what parts of the brain are most active. But you could also look at like where neurotransmitters and receptors for neurotransmitters are in the brain. So for example, this one study, this was at a group at Yale actually, not my group, but another group, did a PET study of, they were looking at smokers and non-smokers, non-smokers and smokers, and actually this was a smoker after abstinence. So I think the actual point of this figure is that they don't look that different. That is not to say smoking's not bad for you. Don't do drugs, kids. But you know, the point is you're looking at, you take tagged nicotine and inject it into the person's bloodstream, and so then wherever the nicotine goes is where the radiation goes, and so these images, kind of the hotter colors, are showing where there's a greater concentration of receptors for the nicotine, so it's showing where the nicotine is actually active in your brain. You can do this with all kinds of different molecules that your brain uses, so that's a cool thing that you can do with PET and SPECT still that you can't do with something like fMRI that just is used for activity. And as you can probably see from here, it's got okay spatial resolution. These images look a little blurry. You know, each kind of pixel is about 4 or 5 millimeters big in real space. But you can see the basic outline of the brain. It's not too bad. And as we said before, it's okay temporal resolution. It takes a few seconds to maybe a minute or two to get these images. So you can't track really fine-grained moments, you know, and what the brain is doing, but you can get a sense of over time what the brain's doing. Okay, so there's a lot of disadvantages too. Temporal and spatial resolution are both worse than fMRI if you're just doing pure activity. It's way more expensive than fMRI. It costs a couple $1,000s, I think, to run one participant in a study, and that's basically because these, well, you know, one of the problems with working with radioactive substances, well, there are many problems, but one of them is that because they're radioactive, they're breaking apart all the time, right? They have a half-life, and they're always decaying. So you can't just like take your radioactive glucose and like stick it in the fridge next to your regular sugar and expect it to be good in like 6 months, right? You have to cook up batches of these radioactive chemicals, and usually they like FedEx overnight. Like if you've got a subject in the morning, I think they like FedEx overnight them, or whatever the special radioactive version of FedEx is that probably costs more. You know, you have to get it by the next morning so all the radiation doesn't dissipate before you actually get to inject it in the person, right? And you've got to make these special substances and so forth. So, and the machine is expensive. It's just all expensive. It's obviously a lot more inconvenient and invasive than a lot of other things, right? Because you're literally getting injected with a radioactive substance. Not enough to like kill you or anything, but you know, it's a concern. And of course, you know, we know what happens when you inject people with radiation. Sometimes good can come of it, not always, right? You know, so you don't want to necessarily start making people into Spider-Man if you can avoid it. So, you know, so you want to avoid radiation if you can. So that's why this is kind of fallen out of favor now that we have other methods for studying pure brain function. Do we have questions about any of that? I thought I saw a hand or two somewhere. Anybody? Pretty good so far. Cool. Okay. So just we'll do do we need to take a break at any point? Also, like we can also take breaks.
Let you go a little bit more, unless you guys do need a break right now, or can you a little bit more? Okay.
Okay. Yeah, just let me know again. Like, feel free to interject because maybe we can go through like everything before like the real fMRI EEG stuff, and then kind of dip into that because I think that's just a little bit more. Okay. So this is more like what you guys talked about this morning, this lesion stuff. So this is real quick, although we could go on all day about people with interesting brain lesions. So this is like possibly the most simple and primitive idea in cognitive neuroscience, right? Is you just wait until someone gets a brain injury, and then see what happened to them, right? So if you want to know what an area of the brain does, wait till someone doesn't have it anymore and see what they can't do, right? So, obviously this is not great when someone gets a brain injury, but for studying human beings, it's all we had for a long time in the real olden days before about 1924, which is when we first started doing EEG in humans. So we've been doing that for almost 100 years now. But before that, you just had to wait until somebody had a stroke, or there were a lot of wars back then, still are, but there were back then for sure. So wait till somebody got a bullet through the head or whatever, and then you could kind of make some guesses as to what the brain area did that was injured, as to what their impairment was. So just a couple of classic case studies. Have you guys talked about any of these people?
We didn't talk about Phineas Gage. He was next on our list.
Oh, okay. Well, I'll just briefly allude to Phineas Gage, and then you can maybe go into more detail later. So this is Phineas Gage. This is have you seen this picture?
I don't have this picture.
This is an actual, I think this was only uncovered a few years ago, an actual photograph of Phineas Gage, who is now I'm doing this all from memory. I don't do my research before talks and refresh stuff, so I might get a couple of minor points wrong, but he was a railroad worker back in the late 1800s sometime, and they would use, I don't know exactly how, never worked on a railroad, so I don't know exactly how this works, but for various things they would pack a certain amount of gunpowder down to a little hole and kind of blast out an area of the ground. And so what you typically do is you would put some gunpowder down and kind of gently tap it with this wooden or this metal bar that you use to kind of pack down the gunpowder, and then you'd tiptoe away and set the gunpowder off, right? So this is one of those tamping rods that you use. And what happened to this guy is he was a railroad worker. He was actually, I think, a foreman. He was pretty high up in the railroad hierarchy. And he was doing this, and accounts vary, I believe, as to exactly how this happened. Someone distracted him or something, but either the gunpowder got set off somehow. Either he packed it down too hard and made a spark, or it got set off while he was still there or something. And this big spiky thing blew up through his head. And this is a simulation based on what we know about him of what happened. So kind of went up through his cheek and his eye socket and came out literally blasted through his head. It landed 50 feet away or something like that. And just took out a big kind of cylindrical chunk of the front part of his brain. Not great. He lived. So this is him after the accident. So it took out his eye, went up like that, kind of took out his eye, but they patched him up. Managed to get his hair over the head hole, I guess, which was good. He's a good-looking guy, so didn't want to disrupt that. But yeah, so this was obviously a major brain injury. It's amazing that he even survived it, right? Because it took a huge chunk of his frontal cortex out. And surprisingly, it didn't actually affect him that much. In the sense of he didn't go blind, he didn't forget how to talk. He didn't forget how to walk. He could still do all of the basic walking around the world functions that we all can. Now, where it gets a little dicey is exactly how much you believe varying accounts. What we do know is his personality changed to a certain extent. His executive function skills definitely declined. There are some accounts that say he became a gambler and a compulsive liar and kind of a sociopath. Very fond of horses, very fond of horses, or just fond of horses, normal fond. I've never heard that one actually. Okay, that's a new one on me. Okay, no, I hadn't heard about the horse thing. I know he moved down to South America for a while, and I think he might have died there if I remember correctly. But yeah, so there's I think he's one of these people that's been built up in the media, and I think there was a kind of shady biographer that tried to make a lot out of him, and it's still a classic study, right? Because it showed that you could who knew you could live without a big chunk of your frontal cortex, and B though he definitely did have some personality changes. And kind of cognitive dysfunctions. It's still an open question as to how much of a psychopath he became after this. But so that's anyway. He's one of the classic studies you always talk about. You guys can probably talk about him more. Do you want to add on to that or? Okay, cool. So another one who we're probably also going to talk about.
We talked about him already.
You already talked about it. All right. So you guys know HM.
Oh, we talked
Oh yeah, so this is from, so he just died a few years ago, as you guys might know it. What's that? 2008. All right, good. So you guys know this. Yeah. So lived up in Hartford Hospital, or at something called the Institute of Living, which is actually, I didn't work with this guy, but that's actually where I worked after my undergrad. That's where I was a research
Did you
assistant.
know he was there?
We did, but like he was, actually he might have been taken away from there by that point and gone to like a kind of older, more intensive, like private place. But for years he was living where I worked. I'm not sure if he was there or not, but I never met him. Marvin Chun, who is not my direct advisor at Yale, but works in the same kind of area, has met him before actually. Says he was very nice. Not great on remembering faces though, as you guys probably talked about. Yeah, so you guys had talked about HM before, had his hippocampus taken out, in theory to help his health, to help his epilepsy, and of course destroyed his memory, or many aspects of his memory in the process. And of course that also gave us classic, amnesia films like Memento and 50 First Dates.
They have not seen Memento.
Oh, really? I mean, it's a little. How old are you guys? Like, what's the age range?
16.
Okay. So I mean, technically, you can't get into R-rated movies yet. So
I mean,
it's 17 what gets you into an R-rated? I can't even remember.
Yeah,
I've not been carded at the movie theater in a while. Okay, so some of you could see it, legally without parental supervision. But yeah, like it's a good movie. I actually haven't watched it in years. But it is a good movie about amnesia. This is maybe a slightly less objectively good movie about, and neither of them is super scientifically accurate, I should say. But still, that's how we got, I think, this amnesia trope of people that can't remember more than a few seconds. Okay, so there are tons of other interesting case studies, but I won't go through all of them. The basic point is, lesion studies do have some advantages. You can show that you absolutely need a brain area for a given function, right? Which you can't necessarily do with fMRI. Like if a brain area activates when you do a task, you know that brain area is doing something, but you don't know that you absolutely need that brain area to do that function, right? So it may be just helping out. It may be just like hanging around being like, hey guys, what's up? I'll activate too, even though I don't really need to be active. That's a little bit of a simplification, but you can actually, with lesion studies, you can demonstrate that like without this brain area, you can't do this function, which you can't really do a lot of other ways. Obviously, there are tons of disadvantages, especially for the subject who would rather keep their whole brain intact. But it's also super imprecise, because it is not ethical or a good idea at all to cause a lesion in a human being deliberately. You don't get to choose where the lesion goes. And it often doesn't affect just a single brain area, right? So stroke often damages a very wide area. If it's a bullet or something like that, you have no idea what the trajectory is going to be. You definitely don't get to predict where the railroad spikes go and so forth. HM was a special case, right? Because they very specifically took out his hippocampus because that's where, they thought the seizures were coming from. But because of HM, we know that we can't do that anymore, right? We can't just take out people's hippocampuses willy-nilly and think that they're going to be okay. So we basically have to wait for nature to provide the lesion, or some people do these kinds of studies in animals, although, it's, again, like you really want to be sure that it's a very important study you're doing before you go around deliberately damaging animal brains to do it, or just be, a really evil person, I guess, if you wanted to, like, go around injuring people's brains. But that's not a good idea. What's that? Yeah. So that's another thing we talked about. We talked about Mengele a little bit in the last podcast. So I mean, but that's part of the reason why we now have like extensive institutional like ethical review, right, of our studies, is because of people like the Nazis who ruined research for all of us. And now we have a lot more. In addition to everything else they did, now we have a lot more paperwork to do these days for. Even a simple like psychological study, you have to do a lot of paperwork for to make sure you're not doing anything evil. Yeah, but yeah, so a lot of disadvantages to the lesion studies, obviously. Okay, and there's really no temporal resolution, right? Like once someone's brain is like before it's damaged, they're fine. After it's damaged, they're not fine, and the brain area effectively never regenerates, although that you may regain some function in various ways. So, there's really no temporal resolution. You can't, with the exception of some stuff we'll talk about in a little bit, you can't really, well, what we'll talk about right now, you can't really lesion and then unlesion a brain. Did you want to break now or do one more topic?
One more topic, because then
Okay.
we get into your stuff.
Okay, so we'll do that. So this is pretty short also. So have you guys ever heard of transcranial magnetic stimulation or transcranial direct current stimulation? Okay.
Is that the one with the wand, or am I thinking of something else?
With the wand? Yeah. You'll see a picture of it in just a second. You were.
Like when you use a little probe and you put in an electrical. It's like
Gotta speak louder, guys.
Yeah, you're all right though. Yes, you are correct that that's what this is. Yeah. So I'll take back what I said about temporary lesions a little bit. So the basic idea of this TMS stuff is, so like we said earlier, your brain activity is really at the heart of it, kind of it is biochemical, but like we said, it's biochemical in a way that voltage is involved, or voltage changes are involved, in the sense that, cells activating or not activating, influences the voltage around them, and conversely, the voltage around the cells influences their probability of firing action potentials. So electricity is tightly interconnected with this, idea of neurotransmitter-based communication. So maybe we know, some of you know, especially if you've done like AP Physics or something, you really know. But I think all of us we all know what an electromagnet is, right? So we know that a moving magnetic field creates an electric field, or conversely, a moving electric field creates a magnetic field. That's called Faraday's principle of electromagnetic induction. So the idea here is that someone said, well, wouldn't it be nice if we could use this principle to influence the electric fields in someone's brain without having to actually go into their brain and inject a voltage with a wire? And you can actually do that. So you take an electromagnet, put it next to a participant's head, and then you manipulate that magnetic field to basically switch the magnetic field off and on in such a way that the changing magnetic field, so it's a sort of convoluted, so you use electricity to make an electromagnet, right? So that creates a magnetic field, and then you change that magnetic field rapidly so that changing magnetic field creates an electrical current in their brain and thus stimulates their neurons. So it looks something like this, if that's what you guys, again, it's a little hard to see. It's kind of like this weird double donut shape. So you put it over the person's head, and you just kind of hold it there, either with your hand or with a clamp or something. And then you don't go inside the person's head at all. It just kind of sits there right next to your head, and you can zap their brain with it. And I finally, after years of being in this field, have you ever gotten this done to you, Sarah? Okay. I finally got TMS'd for the first time while I was in Malaysia. Not for real research, but one of our collaborators in the UK came over, and he was just, he just puts it over your motor cortex, which you guys may have learned is kind of up around here. And if you find the right spot, it'll make your fingers twitch, kind of like that, which was, that was pretty cool. Good to get confirmation that it works and that, doesn't kill you or anything like that. So a very closely related idea, which I'll cover at the same time, is something called transcranial direct current stimulation.
Question.
Oh, yeah.
Go back to the previous slide.
Sorry, what was that?
The previous slide.
Oh, yeah. Yes. Yeah. Oh, just, okay. Any other questions or anything while we're on here? I'll find a way to get you these actual slides, too. So if there's something you missed. Yeah.
What is the use of how do we interpret
Okay, yeah, so we'll get to that more in just a second, but basically the idea is that you can actually use TMS in various ways or in varying patterns of stimulating the brain to either increase activity in the area that it's over or decrease activity in the area it's over. And the idea is that if you inhibit or excite a person's certain brain area while they're doing a task, you might be able to affect how well they're doing on that task, and thus kind of infer that this brain area is involved or necessary for that task. And I have a little bit more detail on that in the slides to come. Anyway, we're kind of building up to that. Any other questions before we go on? So TDCS is very close to the same idea. It's just using a different technology. So it's actually an even simpler idea than TMS. The idea is you just put an electrode on the head, that is, attached to an electric current and deliver, literally like, wire the person up to electricity and deliver current to their brain. It's much like the head-on product for headaches. If you guys have seen this commercial, you literally like apply the current directly to your head. It looks something like this. That's not real. That is not actually what it looks like. That is an artist's image. This is really what it looks like. So I think this is in like an army or military experiment of some sort. So it's just a little array of electrodes that's hooked up to a battery. And it's actually not, you're not just plugging it into the wall socket. It's like a very small voltage. But that voltage basically is able to go all the way through your scalp, all the way through your skull to your brain, and induce these small voltage changes in the area of the brain that these electrodes are over, and either excite or inhibit brain activity in that area. So it's a super simple idea. I'm not recommending you do this, but you could, if you wanted to, like go to Radio Shack and buy the parts to make one of these things for like $50 or something like that. I'm not, because if you do it wrong, you will electrocute yourself. So I'm not saying do it, but I'm just saying you could. Don't actually. One could. You are not allowed to. I hereby forbid you from doing it. But the principle is so simple, yeah, that you totally could. No, don't. So anyway, the idea of both these things is that you deliver current to the brain somehow to either stimulate or increase brain activity or suppress or decrease brain activity. So you can basically use this, especially when you want to decrease activity, to create what people often call a virtual lesion, or like a temporary lesion. So you just temporarily turn off a person's brain for a few milliseconds, maybe a bit longer. And when I say turn off, actually, I don't really mean, we're not just like totally shutting it down. You're really just kind of lowering the amount of activity by a small amount.
So it'd be helpful in epilepsy.
Well, yeah, so in theory, if you could deliver it, so one issue with this stuff is like you can only get at the person from outside the head, right? So you can't, one problem is that you can't target this very specifically, right? And usually, I'm not an expert in epilepsy, you might know more than me, but usually like seizures initiate, I think, in deeper structures in the brain that you might not be able to get to with these methods. So it would be more likely that, if you had severe epilepsy that you couldn't control with medication, that you just get surgery and get actual electrodes implanted in those areas. But yeah, the principle is right that if the seizure were initiating like on the cortical surface somewhere, you could kind of tape one of these permanently to their head and.
If you had one of those where it's like a symptom where you end up like shaking your hand or something.
Yeah, tremors like with Parkinson's. Yeah. So that's another similar situation where, in theory, again, like brain stimulation is a good idea. Parkinson's especially is another one where like the main brain area involved in Parkinson's is very deep inside the head and not on the surface. So usually people do actually do deep brain stimulation for Parkinson's. I don't know if you guys are going to talk about this at all, but okay. So yeah, with that kind of thing, it's a severe enough disorder that like it's worth opening the head all the way up and like going right in the center of the brain and inserting permanent electrodes and doing surgery. Yeah, but for like little psych studies, it's not really worth doing major surgery. So that's why these things are useful. But yeah, in a very limited way, only for really the surface of the brain. That's where most of the interesting psychology stuff happens anyway, is on the cortical surface. Do we have other questions before we go on a little bit? Okay, so yeah, you only can really do this for like, a little short amount of time and a somewhat imprecise, and even like, motor cortex is not too hard to find. It's in the same place for everyone, and I can tell you from experience getting TMS that, just by like eyeballing it, this guy took God only knows what else he was zapping in my brain to like try to find my motor cortex to make my fingers twitch. But he had to zap me 10 or so times to find like the right place to make my fingers twitch, right? Fortunately, I didn't also like pass out or whatever. Nothing else twitched to my knowledge, in my motor cortex. But like, it's not super precise. You're kind of guessing a little bit about where these person's brain areas are going to be. And so the flip side of this is you can use both of these technologies to either suppress or increase brain activity. People have a dream that you can use this as a potential cognitive enhancement method. I don't know if you guys have heard about this. Maybe some of you who like read enough to know kind of what the technology looked like had seen these articles in the press. And people think it might even be something that you could use to treat depression. But actually, there's a company that I found just last night making these slides that sells this stuff. I don't recommend buying this product. That sells a TDCS system that's supposed to help you play video games better, for example. I think it's maybe supposed to be used for other stuff, but clearly this guy is using it to be better at Halo or something. I don't know. I don't. It's like $300. I do not think that is $300 well spent. Probably. That's just my personal opinion. Well, yeah. Yeah, this one hopefully, if it electrocutes you, there's someone you could sue. But like, but even so, like, I don't think. I know that this is actually an area like the military is very interested in cognitive enhancement. You know, people are very interested in whether or not we can use these things to enhance people's brain function for certain activities. So far, it's not super crazy promising in terms of like how much you can really affect brain function, more so than just like practicing more or something, which is, takes more time, but it's a lot less expensive. So we're not quite to like that Matrix level where you can just kind of plug you in and all of a sudden you know Kung Fu. Yeah.
But like in movies such as Limitless, for example, they talk about like how the brain is not being used at its full capacity. So can it go that far as being?
Oh, I have a whole rant about this. That's I could go for days. I should send you. Do any of you guys use Reddit? I sometimes go on Reddit, not as much as more than I should, but less than many people. But I wrote a long rant on one of the Ask Science, which is actually a really good subreddit because it's moderated by experts. So if you have a science question and you don't know an expert, go onto Reddit's Ask Science subreddit and ask real scientists. But I had a whole long rant about this, which I will summarize. But yeah, so first of all, that's like that whole you're only using part of your brain potential thing is not true. You're pretty much using all of your brain all the time. It's more a question of like allocation of resources. So, and this is somewhat speculation, and like what we certainly know is like parts of your brain are not sitting around unused, like vacant lots in a shopping mall, right? So like. That's certainly not the case. Now, we should also mention, like, it's true in a very trivial sense that, like, when people say, like, 10% of your brain, one of the things that might have come from is the fact that only, like, 10% of your brain cells are neural cells, I think. Is that the right figure, roughly speaking?
It's a little.
Maybe it's a little higher. But the majority of your neural cells are actually glia, which are...
Yeah, we have.
Okay, which are kind of support cells that kind of wrap around the neurons and... And of course, you've got stuff like blood vessels and liquid and stuff in your brain that's not biological or that's not part of a cell. So there's all this other stuff in your brain that's not neurons. So neurons are not 100% of your brain volume, but that's not. I don't think that's what people usually mean. Like you just need that stuff to live and have healthy neurons. And we don't need to get into it, but these glial cells may participate in cognitive actions more so than we currently understand. We're not sure about that. Yeah, but this idea that we've just got like unused real estate is not really true. If you look at one of the problems actually with doing neuroimaging is I don't have it in this presentation, but one of our limitations is statistical power, right? You guys probably don't talk to any about stats, right? So you probably get a sense of this though, just from like everyday life, right? Is that you need to collect a certain number of data points to be firm in your conclusions, right? You guys, that makes sense, right? Like so if I want to know a classic example is if I want to know if men or women are taller. If I ask like 5 people, 5 men and 5 women what their height is, there's a lot of variability in height. So maybe I happen to ask 5 tall women and 5 short men, and I find out that women are taller than men in that sample. But that's not generally true, right? Usually men are taller than women. So now if I asked 20,000 people and I randomly sampled them, I just didn't take them all from the country of tall women and the country of short men or something. Yeah, yes. If I didn't only use Amazon women and pygmy men or something like that, assuming I like randomly plucked them from around the world, I would find out very reliably that like, oh yeah, there's this definite height difference between men and women. So that's this idea of sample size, right? And like you can be more confident in your results the more of a sample you have. So this is a really big question. This is a really big issue in neuroimaging because because you need a really strong effect to see these like brain areas activate. And we don't have, because it's expensive, we don't have the luxury of collecting like hundreds of subjects to get all of the more subtle effects that might be there, but we don't have enough power to detect them. So there's one study that some of my collaborators once did where they took a super simple cognitive task that like, it's literally like, you hear these tones in your headphones like beep, 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 and you press a button every time you hear the high beep. And that you just do that for like 5 minutes. And they got 100 people to do that, and they looked at the brain activity, and the whole brain lights up when the high tone activates and you detect that target. And it's like the simplest thing in the world. But yet, clearly, your whole brain is necessary to do that like simple task, right? Maybe not necessary, right? But involved in some way.
Is it also true that your whole brain is active when you're sleeping? Well, not like your whole brain, not the conscious part of you, but like your entire subconscious.
That's a whole. There are many layers to that question. Yeah.
In, like, our last week, so hold on to that question. We'll talk about it then. It's active, but in like an inactive kind of way.
Yeah.
I know that sounds crazy pants, but it does it in such a way that it is active, but it's synced. Like all the parts are kind of synced together, at least during some parts of sleep.
Yeah.
So it's kind of like on repeat.
Yeah.
Yeah, and to follow, I mean, another thing is that like all of your brain is kind of active all of the time, right?
Yeah.
Now, not all parts are equally active all the time, right? So even when a brain area is quote unquote like not doing anything, most cells fire at least a couple of times a second, just seemingly for no reason. Now we don't totally know that it's for no reason, but like if you take a visual cell and you know that visual cell really likes faces and fires a lot every time it sees a face, even if the animal or whatever is not looking at anything at all, that cell will still fire a few times a second. And whether that's just random noise or something else is not always 100% understood. But there is some non-zero level of activity at all times in all cells. So it's really a question of like relative distribution of resources, right? Like so for some tasks, some areas will become a lot more active, and other areas maybe stay the same or even become less active. But nothing's ever really at zero for very long, if that kind of makes sense. And in terms of like memory storage, which is another side of this like 10% of the brain thing, like you kind of use all the brain you have to store all the stuff you know at a certain time with a certain amount of resolution, right? So that sort of means that the more you learn, now you certainly change the connections that you have between your neural cells, but you have a finite number of cells and a finite amount of space in your head. So everything you learn is kind of changing the connections and the weights between the cells that you have, right? So you can sort of change the fineness with which you represent information that may accommodate. So if you like memorize
the cranial nerves.
Okay, sure. So you memorize the
the
cranial nerves. It's not exactly like a hard drive filling up where it's like, oh, well, like because I learned the cranial nerves, that took up 8 kilobytes of space. And because I use an old school floppy disk for a brain, I have to like throw away my high school poetry, to like save room for the cranial nerves or whatever. Just go ahead and throw away your poetry now, guys. It's not going to be as good as your later poetry. No, I'm just kidding. I'm sure some of you are great poets, but many people, I've been told, write bad high school poetry. But like whatever, you don't explicitly like just have to throw away old information every time you learn new information. But it is true that you are sort of like passively, like information from the past is kind of passively fading away all the time if you don't reactivate your memories of it, right? So you are kind of continually reorganizing your resources in such a way to like make the most relevant information the most available, and less relevant stuff kind of gradually fades away over time. But you're kind of using all of your brain for all of that all the time, if that makes sense. There's a whole more we could go into that, but that's a very rough idea. All right, any other questions before we go on? Also, this is another picture from one of those TDCS company websites. Clearly, it'll also make you like sexy and cool looking and like have like what looks like astronomy pictures in the background. Yeah, but yeah. Anyway, all that is to say, I don't recommend um, trying to do cognitive enhancement on yourself with TMS or TDCS. There's all this stuff about neuro-enhancing drugs. That gets dicey too. I mean, they definitely affect how your brain operates, but not necessarily in a good way, right? Like if you have ADHD and you need Ritalin, or Adderall to help treat that, that's one thing, right? That helps your cognitive function. But all these drugs are very dirty, and cognitive enhancement. Your brain is probably working about as well as it ever is going to, just in its normal state. That said, who knows? Maybe in the future we'll get really good TDCS and we'll change our tunes. Okay, so basically just to quickly review the advantages and disadvantages. These are pretty cheap methods to use in terms of what research costs in general. Like I said, TDCS is literally just a few $100, which is very cheap for this kind of stuff. TMS, I think a system costs, I'd have to look it up, but I think about somewhere between $5,000 and $20,000. I think that's a super loose estimate, I know. But in terms of orders of magnitude, that's pretty cheap compared to an fMRI system. It's got good temporal resolution in the sense of you can affect activity on this scale of a few milliseconds to seconds. Okay, spatial resolution, you kind of know where you are in the brain to within maybe a centimeter or so. Not great. You can help that by scanning people's brains and sort of how we talked about earlier, putting the monkey or the rat in a stereotax. Somewhat similarly to that, you can scan the person's brain before you TMS them, look at how their particular brain is organized on the inside, and then figure out what points on the skull correspond to what brain areas and more precisely target the stimulation. You can sort of demonstrate that an area is necessary, like a real lesion. If I zap this part of the brain and someone gets worse at a language task, you can say, well, clearly that part of the brain is necessary to do that language task correctly. But you can't get it in too fine-grained of a manner. So disadvantages are that you do have very limited spatial resolution, and as we kind of talked about, you can only kind of cover the surface part of the brain. You can't get deep because you can only go through that first couple centimeters of skull and scalp and brain surface. You can't use it to tell what areas are active, right? So you're not measuring brain activity, you're only affecting it, and then from behavior, while you're affecting someone's brain, you can kind of intuit what that brain area is doing. So you give somebody a task to do and zap their brain and see if it affects how they do that task. But you can't tell if some other area of the brain elsewhere elsewhere is also active. And it's kind of weird, right? It's kind of messed up. It's not really dangerous. If you don't know what you're doing with TMS, it is, I think, possible to induce a seizure, but with a properly calibrated professional system, not one that you built in your garage, that should not happen. But still, it's not something that you necessarily want to be doing a lot of.
So when you said
Sorry, go ahead.
TMS was used on you and it moved your hand,
Yeah.
did you feel it move your hand, or was it an instinctive response?
It sort of, to me, felt like, so you can, part of it is you can sort of, at least I felt like I could actually feel the kind of, the electric potential on the scalp a little bit, because it is going through your head and everything. So there's a little bit of that, a little bit like you got an electric shock on your head where it is. But to me, it sort of felt like if you ever have gotten a strong static shock in the wintertime or something, and makes your hand kind of twitch away from it or something, it sort of felt like that, except without any external cause. You know what I mean? So it's sort of like your finger just all of a sudden twitches and kind of stings a little bit, but not in a painful way, just kind of in a, it's an odd sensation, but you don't.
Because you didn't actually think about doing it first.
Yeah, it didn't feel like thinking about moving my finger. You know what I mean? It felt like a reflex twitch or a muscle spasm, kind of. But yeah.
I don't know if you've heard, but there's that one thing about muscle memory. I did this when I was little, but you go stand in a doorway and you push your arms against the side, and then when it involuntarily raises your arm, was it like that, or was it different?
It's sort of like that, I guess, in a way that it. Because you guys know that thing that everybody has done that before, where you put your arms in a doorway and push up with your arms. Everybody know what I'm talking about? Nod. Yes, no. You sort of go to a doorway.
You, Matt.
Yes. And can all do this during the break. You like push your arms up like really hard, and obviously they can't move up because your arms are in the doorway. And you do this for, I don't know, like 15, 20, 30 seconds or something like that. And then you walk out from the doorway, and your arms just kind of like float up in the air magically. Now, the thing about that though is, right, you've done it before, so you know that like if you think about it, you can keep your arms at your sides, right? Like it's just that it feels like the amount of muscle tension you need to just let your arms hang is actually the, like, you know what I mean? Like that amount of, like what feels like you feel like you need to kind of push your arms down to keep them just hanging, which is, it's weird, right? But like it's not like you can't possibly keep your arms down. So it's a little bit different, like in the sense of like you can't stop the twitch when it happens. But I mean, it's also like it's like that fast, right? It's just like a little like instantaneous thing. So it doesn't. It also makes a sound, like a little kind of snapping sound. So
Like, is it just in your brain or is it like outside?
well, I mean, it's stimulating. You mean the TMS or the doorway thing or both?
The TMS.
Okay, so the TMS is directly stimulating your motor neurons, which, I mean, they're part of the brain, but like they start off here and then they go all the way down. Basically, this is going to test my neuroanatomy. They go all the way down to your spinal cord and have, I believe, one synapse, right, that then goes out to your arm and connects directly to the muscle. So it's a nearly direct connection to the muscle, right? It's only like kind of one synapse. So like the motor neuron synapses in the spinal cord. That neuron has a synapse with your muscle, and then your muscle twitches, right? So it's like a very, it's almost like a reflex that goes like to your spinal cord and back. Like when you get a shock and your hand twitches before you're conscious of it. It's almost that automatic kind of. It's not very cognitive. It doesn't feel like much until after it's happened. You know what I mean?
Okay, let's take a break.
Okay.
Okay. 10 minutes. So.
All right, so I guess we will start up again here. Oh, look, we are on functional MRI now, which is my favorite. It's my baby. Yeah, so once again, like we're getting into more of like my actual stuff here, but still like, so we'll get into a little bit more detail with this stuff. Still feel free to ask me questions and stuff, because again, I can go really as long or as short on this stuff as you like. If we want to just turn this into an all-nighter and I can like teach you all about fMRI, we'll do that. Or if you just want to like, come back to the mini Yale and work with us for 4 or 5 years in the lab, you'll really get to know it. Yeah, because this stuff is really complicated, but I'm going to try to boil it down for you as much as possible. It gets the physics and stuff get a little hairy. One thing I have told, like we've had some undergrads and stuff working in our labs recently, and one of the weird things about cognitive neuroscience, and all neuroscience, and like neuroscience is very interdisciplinary anyway, but I think like if you want to be really good at cognitive neuroscience in particular, you have to know like psychology, you have to know physics, because like there's a lot of physics in fMRI and electromagnetism and stuff like that. You've got to know some chemistry and some low-level cellular type neuroscience to kind of know what's driving the effects you see. You got to be a pretty good computer programmer because there's a lot of programming involved. You've got to be a statistician. You've got to know like 10 different things. So it's a kind of jack of all trades sort of discipline. It takes a long time to learn it all really well, but I will try to give you the gist of it now. Okay, so little haha. So you guys, this is not that funny of a comic strip, really. This is from XKCD, by the way. If you guys are not big nerd comic readers, all of my comics, I think, come from XKCD, Saturday Morning Breakfast Cereal, or Abstruse Goose, which are all of the good nerd comics for if you give a lot of lectures and need nerdy comics. But you guys have probably all seen, we've actually already seen this with PET, like images in the news or whatever that show a little blob of activity, or like this part of the brain is for this and blah blah blah. And usually those sorts of images come from fMRI. This one is obviously just drawn, but these like this idea that you've got a little blob of activation or a little brain area that activates, and that this is associated with some cognitive process, comes from mostly fMRI these days. Now we'll talk a little bit about how that's not exactly the full story. It's not at all the full story, but first we'll get into how it does that in the first place. So fMRI, so any of you guys had an MRI before for like a knee or something, a sports injury? Okay, a lot of people have had MRIs. So fMRI is just an application of MRI. It's a way that you can use an MRI scanner. So it doesn't, F stands for functional. It doesn't measure neural activity directly. It's a very indirect measure of brain activity. So instead, it relies on this principle that we've sort of alluded to earlier with PET and stuff, that when you use a brain area, you get an increased blood flow to that area, right? Now, we've actually known this basic idea. This was one of my favorite little cool things to show for over 100 years. This slide, full disclosure, is like completely stolen from some other PowerPoint I found on the internet like 1,000 years ago. I have no idea whose it is. So if you're out there, person, thank you for your slide, which I have just copied in full. But basically, their version of the first quote-unquote brain imaging experiment, there was an Italian researcher named Angelo Mosso. This is reported in William James's Principles of Psychology, which was like, William James was one of the first and most important American real psychologists. And this book, Principles of Psychology, is like, people quote it all the time because I actually quote it later on, because it just had like all of these ideas that we've been working on for like 100 years, kind of. He was very prescient, very ahead of his time with a lot of the ideas in this book. But anyway, he tells the story of this guy Mosso's experiments, where his version of quote-unquote brain imaging was to get a person to lie very, very still on a very exquisitely balanced board, so that the person was like perfectly balanced front to back, and then have the person thinking, in this case he said the moment emotional or intellectual activity began, so get the person to think really hard somehow. I'm not sure exactly how he did this, or how he evoked emotional activity, but at least in theory, according to this, according to the lore of this experiment, when the person's brain became more active, a little bit more blood would flow into the brain, and the balance board would like tip down, towards the head and away from the feet. And that showed that, thinking caused this rush of blood to the head. Now, if we go back to our little chart of like, advantages and disadvantages. Advantages, very cheap. You can build this for even cheaper than you can build like a TDCS system. Disadvantages, not great spatial or temporal resolution. You know that the activity is located somewhere in the top half of the body. But that's about all you can say, right? And then like, not a lot of temporal resolution either, because like once the board tips down, I don't think you can like make the blood rush back to your feet to like, set it right again. So not very useful for most things, but like the basic principle we are still using with fMRI these days. I'm not totally convinced that really works. I've never tried it myself, but maybe if somebody wants to make me a Kickstarter, I will totally build one of those things and try it out. Do like a Mythbusters. Okay, so
You guys could do it for your project.
you could totally do that. It doesn't take that much hardware. So like I said, I can't really explain everything about how fMRI works, so let me try to just sum up as briefly as possible without being like totally inaccurate. There's a lot of, like I said, there's a lot of physics involved. Most people that even do this for a living don't like really know all the physics. I don't know all the physics. I know more than some, less than others. But I'll give you like just a taste of it. And if that's your thing, we can always talk more about that later. So like I said, functional MRI is just one type of MRI you can do. Use the same machine for all of it. It's based on the idea of nuclear magnetic resonance. I don't know if if people have taken like AP chemistry, you might have talked about that. I'm not sure. We don't need to get. Okay, it's not okay. Maybe people have heard of it. Have you guys heard of it? Maybe okay. So NMR is a way that you can like sort of tell what a chemical compound is or look at properties of it. So it's based on nuclear magnetic resonance, but when they named MRI and they started using it in hospitals and stuff, they took out the word nuclear so people would not think people would not worry about the radiation element, because it doesn't use any actual like harmful radiation. Ironically, unlike PET, which sounds like very cuddly and like, oh, it's just PET, and really they're like injecting you with plutonium or whatever. It's not plutonium usually; it's like radioactive carbon or oxygen or something. But still, fMRI, there's no radiation in that sense. So that's unlike PET and SPECT or X-rays, which obviously use X-ray radiation, or CT scans, which are just basically fancy X-rays. None of that type of radiation is involved with MRI. It uses a totally different type of physics to get its images. You do use quote-unquote radiation in the sense that all light waves and radio waves and everything else is a type of radiation. but it's not the type that is harmful. So it uses regular radio waves and a really strong magnet. And I mean, that's not really all you need. You need a lot of computers and a lot of other kind of supplemental equipment, but the basic principle is just a magnet and some radio waves. So the way it works, if anyone has had an MRI before, you know that you have to put your person in a really strong magnetic field. Anyone Breaking Bad watcher? So you know, think of the scene where they broke into they tried to get the laptop out of the jail, and they got the junkyard magnet, and so it's a really strong magnet on the order of that strong. So you have to take your keys out of your pocket and stuff before you go in the magnetic field. So how strong is it? It is actually, so they measure magnetic fields in Tesla, named after Nikola Tesla, who you guys may have heard of, the dashingly handsome Nikola Tesla, who ironically I think loved science so much that he never married or anything, despite his dapper nature. So the strong magnetic fields are measured in Tesla, named after this guy who did all kinds of crazy. He invented the alternating current motor, which is like kind of important. But he also did like all these crazy experiments and thought he could split the world in half. He was a little bit nuts. But we named the unit after him. So to give you an idea of how strong the magnetic field in an MRI is, the Earth's magnetic field, like the kind that drives your compass, it varies from place to place, but on average it's around 0.00005 Tesla. So that's pretty, I mean, it exists. It's enough to turn a compass needle, but it's pretty weak. You don't really, because of the, you don't find your keys like flying off towards the North Pole or the South Pole like on an everyday basis, right? So it's pretty weak. That junkyard magnet, like the one that they used to pick up cars in a junkyard, or the one that they used in Breaking Bad, that's between 1 and 2 Tesla usually. And the magnet that you go into to get your brain imaged or some other body part imaged, depending on the one you go into, and like a hospital, sometimes it's only 1.5. For good neuroimaging experience, usually now it's 3, but sometimes up to 4 Tesla. They've got MRI scanners that go up to at least 11 or 12 Tesla. So they're really, really strong magnets. And I think the strongest, I was just reading the other day, the strongest magnetic field ever created by man that was stable, like that didn't destroy itself. Because I guess you can make a stronger magnet, but it rips itself apart, is like 100 Tesla. But you could not put a person in it. That would have to be a very small magnet. So the smallest, or the strongest person-sized magnet, I think, is around the 11 or 12 Tesla, but it could be higher now. Anyway, so yeah, and this guy with his magnetic personality, 1 Tesla. All right, so that's how strong it is. So it's literally as strong as one of those junkyard magnets, or stronger, is how strong this magnetic field is. So what this does when you go in there, fortunately, we are not made of metal. So when you go in there, you do not stick to the side of the magnet. What it does do is it affects the atoms in your body at a very low level, or actually the protons in the atoms in your body at a low kind of physical level. So, and this is where the physics get kind of weird and complicated, probably beyond what's worth talking about. But basically, protons have a spin. So we talk about subatomic particles as having a spin. So you can imagine them as being like little whirling tops. And that spin gives them their own little magnetic field. So like, if it's spinning around clockwise, one side, so the axis around which it is spinning, one side of that axis is like its north pole of its little magnet, and one side is its little south pole. So you can imagine this just like, we talked about electromagnetic induction, right? So like moving electrical field creates a magnetic field, right? So if you have an electrical field moving around like this, like the charge in a proton, that causes a magnetic field, right? So charge in the proton is spinning around, that creates a little magnetic field. I think I know enough particle physics to know that that's not really good or accurate way of describing it, but you can think of it that way. So each proton is like a tiny little bar magnet. Putting you in that magnetic field aligns your protons with the field. Then what you do is you hit the person with a burst of radio waves. And like these are actual, so it's a car radio. These are actually just real radio waves like you would have in your car radio. So in our scanner that we use at Yale, our 3 Tesla scanner, the actual frequency used is a 127 megahertz. So that's actually a little bit higher than what's on like your FM radio dial. So these numbers stand for megahertz. So it would be just like a little further out. But it's the same basic type of say it's literally just radio waves, like you would use, to power a radio or a TV signal or whatever. I think the actual frequency that's used in MRIs, I looked it up, and like the spectrum is all assigned to different things, like certain microwave frequencies are for your cell phone and all this other stuff. So that frequency is apparently assigned for some sort of aeronautical navigation. So like probably you don't want to take your MRI on board a plane just in case that crashes the plane's navigation systems or something. But yeah, so that's the frequency used. But it's just regular radio waves of that sort. So I have a little. So I totally stole this from some textbook at some point, and I think the website has since gone offline, but I fortunately made a copy of it ahead of time. So just to give you like a little kind of visual aid as to what's going through. So that's what an MRI scanner looks like. I'll steal their nice little sexy proton figures. So this is, like I said, you can kind of imagine the proton this way. So it's got a little positive charge. It spins around. That creates a little magnetic field, a north and a south pole. So that's just its spin or its angular momentum. So normally, you got a lot of protons in you. Those of you who know some chemistry, what do you call a single proton by itself could also be, well, by itself with maybe one electron also, would also be called what? Hydrogen, right? So yes, that's what you said, right? Yeah. So hydrogen, right? So you got a lot of hydrogen in your body. Hydrogen's kind of important. It's in all of your organic molecules and so forth. So every little bit of hydrogen you've got in every cell and every molecule in your body is one of these little guys. So you got a lot of them. Especially, what is the thing that you have most of in your body? Okay, excellent, very good. So especially, every molecule of water you have has 2 hydrogens in it, right? So all of those hydrogens are normally just kind of like lying around, unorganized. But then when you put them in the magnetic field, they kind of jump to attention like this. Now, one thing to note is that they line up with the magnetic field, but they don't all line up in the same direction. So they can either line up kind of one way with the magnetic field or exactly the opposite way, right? So like some are kind of standing upright, some are kind of lying down this way. But they go one of those 2 directions in alignment with the magnetic field. And then this is where I'm probably, that's what happens when you just put them into the magnetic field. And then what's going to happen is when you hit them with a burst of a radio wave, I can't remember, at a certain point this gets more complicated than we want to care about. But when you hit them with a radio wave, these protons are going to absorb some of the energy from that radio wave. And basically what's going on is when the protons are perfectly aligned with the magnetic field, that's a lower energy state. So you can think of it this way. Like, think of yourself standing upright on your feet, right? Like, that's a relatively low energy state, right? Like, you can maintain this without a whole lot of effort. And you've got, in this case, the field that you're aligning yourself with is like gravity, right? So like, it's easy to align yourself with the gravitational field of the Earth. Now, if you were to stand on your hands, that would be harder, right? It would be possible to maintain your balance and so forth, but like, it would be harder. It would take more energy and effort to, like, you know what I mean? That would be a higher energy state. That would be a more difficult state to maintain. But both of those are easier to maintain than like standing at a 45-degree angle, right? Which you can't really do at all, right? So these protons in the magnetic field, some of them choose to like stand on their feet. In fact, a little bit more of them choose to stand on their feet and occupy this somewhat lower energy state. But some of them are standing on their hands on, in this higher energy state. But they're all in one of those 2 orientations. None of them are kind of like leaning at a 45-degree angle. Okay, so that's the idea. And then it takes more energy to kind of maintain the high energy state than it does the low energy state. So that's why more of them want to be, just like you guys, most of you, when you're just chilling, are standing on your feet and not your hands, I assume. So as far as I've seen so far. So like, you most of the time choose to occupy the lower energy state if you can. Okay, so now I'm going to skip away from that diagram because that gets into the real details, which are annoying and complicated. So this is the basic idea, right? So now, again, if you guys have done any chemistry or physics, what you might know is that when you add energy to something, you change its state in some way. So it has to do something with that energy. It's like, it's like you drink 5 cups of coffee. You don't just like sit there. You twitch, right? You occupy. Maybe then you're like, if I give you guys enough coffee, you will start walking on your hands, right, just to burn off the energy. So it's sort of like that. Like, if this proton absorbs this energy from this radio wave, it has to go somewhere. It has to change the state of the proton in some way so that the proton uses that energy. It can't just be like, thanks for the energy, I'm not going to do anything with it, because that ruins the conservation of energy. So what happens is if it absorbs one of these radio waves, the proton will flip. One of the lower energy protons might flip upside down into the higher energy state, right? So then what's going to happen, though, is after a while, again, this is anthropomorphizing the protons a little bit more, but they get tired. They don't want to stand on their hands anymore. They want to go back to their lower energy state. So they flip back down, but then they need to release that energy, right? So again, if they go back to a lower energy state, that energy has to go somewhere. So what they're going to do is emit their own little tiny radio wave to give back the energy, send it back out into the universe, because again, that energy has to go somewhere. And basically, we can detect this radio wave. So all you need to detect the emission of a radio wave is an antenna, right? So like that's how your car works. It's just the antenna is just a piece of wire that sucks out radio waves from the air. So essentially, that's what we're doing is we're first hitting all the protons in your body with this radio wave, then waiting for the ones that kind of flipped into their waiting for the protons that flipped into the higher energy state to get tired and flip back down. When they do, they send out these little echo waves. We call them echoes because it's like send it out and it comes back. And that echo wave measurement is the basis of our fMRI signal. Now, the details get really complicated, if that wasn't complicated already. But that's the basic idea, right? Is that we can detect basically an echo of what we sent out. So we're kind of doing this thing where we momentarily perturb the protons and then wait to hear the echo back. Does that kind of make sense? Do people have questions about that? I know at this level of analysis, a lot of it still maybe doesn't make that much sense. Yeah?
So the radio wave gives the protons some energy that
Yep.
makes them flip up?
Yes.
I thought photons that use that.
Well, so a radio wave is a photon, in fact. So photons are, so if we want to get into it, yeah. So again, this sort of depends on how much physics you guys have done. So everything that involves electricity, magnetism, and light, any kind of light type radiation is part of the electromagnet. So there's 4 fundamental forces in the universe, 2 nuclear forces. this is neuroscience going very low level. There's gravity, and there's 2 types of nuclear forces: the strong and weak nuclear force, yeah, and then electromagnetism, right? So although it does not seem that way, right? Like electricity, magnetism, and light are all kind of the same thing. And basically, like all of your radio waves, your gamma rays, your X-rays, your visible light, your infrared radiation, UV, yeah, all of that stuff are all just different energy levels of photons. So you're right. So when you hit something with a photon, basically that, when you hit a matter particle like a proton with a photon, that gives it energy. Yeah.
Someone's told me that's why there's a limited speed of light, because they're particles, they're tiny particles. Sure, they can move like really, really fast, but still they can only go finitely fast because they're particles. If that makes sense.
Yeah, and they have, this isn't a topic that comes up all the time on the Reddit Ask Science thread. Yeah, I mean, and there are other ways of thinking about it, like photons are massless, right, or thought to be massless.
Yeah.
And the amount of mass you have kind of, if you have any mass, you can't go the speed of light. But it gets into this whole relativity thing, right, how like space and time are related. Yeah, let's not go down that road. But the idea is that you can sort of, the whole thing of like if you go faster, time slows down for you. The idea is sort of that you can travel a certain amount through space and or time, right? But if you travel through space faster, you travel through time slower. Anyway, but let's not get into that. Any other questions that don't involve like totally explaining the universe? No, although like I can totally go down that road, but we probably should not for time's sake. Okay, so anyway. So we get these little radio wave echoes from our, again, you can sort of think of this as just like what a cell phone tower does with your cell phone, right? Like it sends out a big burst of, in this case, microwave radiation that hits the antenna in your cell phone, and then your cell phone does some stuff, right? And then sends a little bit of a little teeny echo back that larger antenna is able to pick up again, right? It's the same kind of idea. A much bigger antenna hits your head with a big burst of radio waves. Then each little proton sends out a little baby echo of that original signal at some point. And you can detect that, right? So you have kind of this two-way signal. Okay, so the thing that makes MRI interesting, if that's all that happened, like we would just get like a whole glowing head wherever there were protons, right? And there's protons everywhere, all in the air and all through your body. So that's not that interesting. So the interesting thing is that various factors about the chemical composition of your body tissues affect the strength and timing of that echo signal. So basically the idea is that your body composition, each molecule has its own kind of electrical properties and its own shape. So that kind of very slightly deforms the magnetic field. So there's this sort of basic, big, strong magnetic field we're pumping in from the big magnet. But then, your local tissue kind of distorts that magnetic field. And those distortions lead to small variations in the strength and the timing of those signals we pick up from your protons. So by looking at exactly what types of echo signals we get back, we can therefore conclude something about that'll affect the brightness sort of the image that we register. And we can conclude stuff about what types of tissues those signals are coming from, okay? So again, this gets very complicated. This is a whole area of research unto itself, but what I haven't told you about is all the various ways that we can adjust exactly what this radio wave is that we send out, like how strong it is, how frequently we send it out. And we can adjust our magnetic field. So in addition to the sort of big magnetic field we put you in to line up all your protons, we can also kind of tweak the magnetic field in all three dimensions, and add small deviations to kind of, that helps us tell where in the brain these echoes are coming from, or where in the body period these echoes are coming from. So we can tweak all these little things about it that kind of go beyond the basic physics of MRI. The basic idea is that the combination of how we tweak how the scanner works and the different properties of the tissues in your body leads to us getting like slightly different strength and differently timed signals back. And that lets us figure out what the tissue is and where in space it's coming from. Okay, so how we figure that out is probably beyond the scope of this lecture. But I will tell you that fMRI depends on a certain type of MRI signal, which is called BOLD, which stands for blood oxygenation level dependent signal. And the idea behind BOLD is that oxygenated blood shows up brighter than deoxygenated blood. So you guys know that your blood carries oxygen to your body tissues. When the oxygen gets used, the blood is deoxygenated, has to go back to your lungs to get more oxygen. Do you guys know what molecule in blood carries the oxygen? Okay, good. Yeah, you guys are probably, hand raisers are gonna just shout at me. That's fine. Everyone can shout. Yeah, so hemoglobin is and everyone, if you have questions, you can also just shout at me. That's why I'm not good at detecting raised hands. So hemoglobin carries the oxygen. Does anybody know what element in hemoglobin is really important that is not in most biological? Just shout. Iron. Good. Excellent. Right. And if you guys saw the first X-Men movie, you can there was that whole thing where he sucked the iron out of the guy's blood, right? Although that guy had extra iron injected into him, I think. Magneto sucked out the iron from the blood. Fortunately, an MRI scanner is still not strong enough to suck the iron out of your blood. It's strong, but it's not that strong. But anyway, that iron in your hemoglobin, basically what happens is when oxygen binds to that hemoglobin and it becomes oxygenated, iron, if you guys have ever used a magnet before, iron is magnetic, right? So when that oxygen is bound or not bound to the hemoglobin affects how magnetic this whole molecule is with the iron in it. And that essentially leads downstream to a difference in your MRI signal according to whether the blood is oxygenated or deoxygenated. So anyway, all that is to say is oxygenated blood is brighter than deoxygenated. Do you have a question?
Does it show the blood flow?
Is it what?
Does it show the blood flow?
Yes. So actually, this is one thing to kind of mention is although we talk about blood oxygenation level dependent signal, Actually, most of what we're measuring with fMRI is just the sheer amount of blood flow, not how oxygenated it is. So we'll get to that in like just 1 second, actually. But let me know if you have more questions about that.
One
Oh yeah, where? Or
question.
just ask.
On
Oh,
the
yeah.
other side, I still understand how the oxygenated blood shows brighter.
So this you're getting dangerously close to the part that I don't remember. But the basic idea is that so materials can be either magnetic, so like iron, just like pure iron, or I think it's nickel and magnesium are the only three elements that are like just straight attracted to a magnet, right? So like if you put a magnet next to a chunk of iron, it sticks to the magnet. Most materials are not magnetic in that way, right? Even other metals. So if you put a magnet next to aluminum, it doesn't do that, right? But most metals are what's called paramagnetic, which means they're affected by the magnetic field, and it does stuff to the metals. And then again, my physics knowledge starts to break down at a certain point here. But basically, they're affected by the magnetic field, but they're not attracted to it exactly. So like if you take a quarter, you're not supposed to do this, but I know what happens. I can't tell you how I know this. But if you take a quarter and like stand it up inside an MRI scanner, it'll just stand on edge forever without falling down because the magnetic field doesn't like pull the quarter to the magnet, but it does like exert a force on the whatever metal is in a quarter that kind of keeps it upright. So you've got paramagnetic and then I think and then you've got things that are not affected by magnetism at all or less. So anyway. Basically, the idea is that when you bind oxygen to this hemoglobin molecule or don't, and then I don't know enough physics or chemistry to know exactly how this is, you change the properties of that whole molecule from being more affected by the magnetic field to not as affected by the magnetic field. So it changes from paramagnetic to, I believe, not at all magnetic. And anyway, in essence, what this means is, like we said, different. You can think of it as like the magnetic field that we induce with the MRI machine kind of deforms around different tissues in different ways. So different tissues, they're all filled with their own protons and electrons and, have stuff moving around in them that all have their own tiny little magnetic fields that are both affecting and affected by that bigger magnetic field, right? So like we kind of create these tiny little deformations in the field. And essentially, what it means is that oxygenated hemoglobin and deoxygenated hemoglobin deform the field to different amounts. And anyway, what that means is like then that has an effect on nearby protons and how they're affected by that radio pulse, and thus the strength of the echo they send back. But it gets more complicated, and like I'd have to remind myself of how some of this. It's the kind of thing that you like every couple of years you refresh your memory and then you forget 90% of it because it's you don't have to do this to like do the work most of the time. Is that enough of an explanation to kind of, sort of? Yeah, it's a whole thing. But I know that's like more physics than probably anybody wanted today. But okay, any other questions? Is everybody sort of on board still? Like I said, you got to be a little of everything to know what you're doing here. Anyway, so getting back to like real what's happening inside the head in the real neuroscience, what really happens is when a part of your brain activates. What essentially happens then is there's this tiny little dip in your fMRI signal because a little bit of oxygen gets used up and your hemoglobin becomes deoxygenated. That gives a small decrease in the MRI signal, but that's actually very small and very brief. And it's very small compared to what happens next, which is that for reasons that we don't fully understand, your circulatory system like hugely overcompensates for the amount of oxygen you use and sends like a big influx of blood in to replace this tiny little bit of oxygen that you used. So the amount of blood that comes flowing in afterwards is like way more than the amount of oxygen you used up in the first place. But it's good for us with MRI because it lets us do fMRI. We don't exactly know why that happens though. And by the way, when we say large, it's not like every time you do a math problem, your head does not like swell to twice its size. So we're really talking about like a 1%, like if you get a 1% signal change in an fMRI study, like you feel pretty good about yourself. So that's large is always a matter of perspective, I guess. But that's still much larger than the amount of decrease you see in the signal from the loss of oxygen in the blood. Right. So anyway, although we say fMRI signal is oxygenation dependent, what we really care about more is how much blood comes flowing in after you use your brain. And so basically what we try to do when we do MRI is we put the person in the scanner, start the scanner up, give them something to do with their brains, give them a cognitive task of some sort, and we say, all right, we know when certain cognitive events are occurring because we're making them happen. We're controlling the timing of the task. And we know from previous research what basically the signal in your brain should look like when you've used an area of your brain. So what we're going to try to do is find areas of the brain where the fMRI signal matches what we would expect if your brain activated at certain times. And if those correlate closely enough, we say that that area is active in our task. So I have a quick little demo to kind of illustrate this for you. So let me go back to MATLAB. Probably nobody here has used MATLAB before. Anybody? Probably not. You guys are not at the level where you'd have to. It's a scientific computing program. But this was like the biggest day of my life. I drove by the MATLAB headquarters on the way because it's in like Natick, Massachusetts. Is it Natick? Is that how you say it? Natick. Yeah. So like whatever route the Apple Maps GPS took me by the MATLAB headquarters, and I was like, oh, it was awesome. Like probably not for you guys, but it was pretty great for me because I have been using MATLAB like 6 hours a day for the last decade or whatever. So anyway, I'm going to give you a little demo of what we call the a little demo of what we call the hemodynamic response function. So this is what we're gonna see is a little chart of what the sorry. Just gonna quickly edit my code a little bit. I have a different version of a piece of software on this machine. Okay, so what you're gonna see here is a little demo of what the fMRI signal looks like for a period of time after an area of your brain is activated. Okay, so let's imagine I stimulate your brain in some way. Or let's have you stimulate your own brains in a sense. So when I give you the signal, so let's say when I clap my hands, shake your own hands like this for about 2 seconds. Okay, just give me jazz hands for like 2 seconds, and then what we'll do is pretend that we're recording fMRI activity from the motor cortex of your brain that controls your hand movements. Okay, everybody ready for that? All right, this will wake you up too. All right, I think I remember which key triggers the demo. And what you're going to see is a real-time plot of the fMRI signal in your motor cortex if we were scanning you. All right, and go. Jazz hands, jazz hands. All right, so here we go. And so like you can stop jazz handsing now. So this is what's going on in your brain. Is like the blood flow peaked a couple of seconds ago. It's now coming back to baseline in your motor cortex. And what it's actually going to do is it's then going to actually kind of go below the sort of resting state level for a little while, and then like gradually recover, assuming you don't move your hands again. It's going to gradually recover to sort of back to its resting state. So this is in real time, and this is kind of an ideal model, but this is approximately what happened in your brain when you clap your hands in your motor cortex. And what you hopefully noticed is that this whole process is super slow, right? That took about 30 seconds for it to go, for the signal and thus the blood flow in that part of your brain to go up to a sort of peak amount, decrease, and then like kind of recover back to where it was. So it takes about, even if you're just worried about like the maximal change in signal, which is right here, it takes about 5 seconds. So like it's about 5 seconds after that part of your brain was active that we actually see the biggest change in the MRI signal, even after that activity is gone, right? So it takes about 5 seconds for that flow of blood to kind of rush in to that part of the brain. So that really gives us some challenges with MRI if we don't see the results of your brain activating for several seconds, after it activated, right? Now, just to give you an idea, sorry, I'm doing the wrong thing here. Just to give you an idea of when we do MRI, we actually can't signal this. We can't sample the signal this quickly. So we can actually only get an MRI image, depending on how we do it, about once every second. So actually, these little points I highlighted here are actually what we would record in that part of your brain. And so if you take the sort of idealized plot away, that's actually what we'd measure. Is we'd measure this amount at time 0, this amount at time 1, this amount at time 2, this amount at time 3. Or actually, this is I think every 2 seconds. So time 0, time 2, time 4, time 6. So we might be able to get a few more dots than this depending on how we set up our scanner. But this is what we're basing our estimates on to determine that this part of your brain was active at time 0, right? And of course, that's assuming you didn't do anything else with that part of your brain in that intervening 30 seconds. Because if you did, you'd start that whole cycle over again, and you get kind of this weird additive signal that would be hard to disentangle, right? So this makes life complicated for fMRI people. Before we go back to the slides, does that kind of make sense? Anybody got questions about the basic idea here? So the basic upshot is it takes about 5 seconds for that MRI signal to reach its peak and about 30 seconds for it to totally recover, which means that even if we can run our MRI scanner and get images every 1 or 2 seconds, like your body is so, that's what I was talking earlier about your dumb, stupid capillaries and your stupid brain, limit how useful this technique is to us, because no matter how fast we can run the machine, your circulatory system in your brain only works so fast. Isn't it? So we have gotten a lot better. Just to give you an idea of like what we do see with this technology, we've gotten a lot better over the years. So I found somewhere sometime one of the first, this might be the first image from MRI in the 70s when it was first developed. This is not taken with the scanner. This is a photograph of the guy in the scanner. He does not look that happy to be there. I think this is probably one of the graduate students or postdocs on the project. Also the test subject. So this was how they did it at the time. They just had this big coil that like literally went around his chest. They didn't have these nice hospital quality machines back then. And that was the image they got out of that. And it's sort of like, if any of you guys played the old school like Nintendos or Ataris, sort of like 8-bit art of the human body. It's like I think this is the heart and these are probably the lungs, and that's just like, everything else. So this is like a slice taken. Do you guys know the anatomical terms yet? Okay, so this is now I'm going to screw it up. An axial slice through his...
Otherwise known as horizontal.
Right. I had to actually think for a second because it's different for some of the planes in the body versus the head, and I'm only used to the ones in the head. Because I'm not going to get up on the desk like a dog and demonstrate, but like all the anatomical terms were based on the idea that, we're quadrupeds and like, that our spinal cords, like the spinal cord goes like different, in a dog the spinal cord goes that way, and a human it goes this way. Anyway, so that's a cross-section through his chest, and I think that's his heart and his lungs and stuff, but you can't see a whole lot. But then again, that was when they first invented this stuff, so it was pretty coarse. Now, in comparison, this is a brain image taken earlier this year in my own research. Got a lot more detail, you can see. Now, not all images are going to look this good. You might notice that this is an exquisitely well-formed and handsome brain. That's because it is my brain, and that is from a study that I was both the runner of and the participant in earlier this year. But that's a pretty clear image of my brain, actually, as things go. And just to give you an idea, so that was a structural brain image, so that takes a lot longer to acquire to get that level of detail. We're talking about functional images that actually measure brain activity. And sorry, this again is going to come up like overly bright on this projector. But basically, this is the kind of quality we were getting on a functional brain image in like 2006. Pretty blurry. These are all images from my real research. So you can see by 2010, we'd improved things quite a bit. We got a lot more detail, got a lot more contrast. You can actually see like differences in the structures there. And if we slide that over and show one from this year, we've gotten even better. So you can get a lot more detail, a lot more contrast, and a lot better signal to noise. So we are getting like better and better all the time. These are actually, probably means nothing to you guys, but like for people in the field, these are like gorgeous brain images. I think this one's also me, actually. I think these ones were strangers, but this one's me. So it's a really beautiful brain image, and it only takes us about 1 second to collect this image as compared to, 8 minutes to collect the really high detail one I showed you a minute ago. Okay, so a few other just like random facts and figures about MRI and fMRI. Like I said, that structural brain image, which in other words doesn't measure activity, it just kind of shows you like, here's all the structures in the brain, here's the white matter, here's the gray matter, here's all the empty space, hopefully not too much in my brain, but you never know. That just to kind of show you where the basic anatomy is. Those pixels are actually, we call them voxels. So a pixel, like on a computer screen, is a picture element, but if it's in 3D, it's a volume element. That's where the word picture element becomes pixel, voxel, volume element becomes voxel. So the voxels, or the 3D pixels in our brain images, that was 1 millimeter wide, was each pixel in that image of my brain. So that's about the spatial resolution we can expect. And like I said, that takes about 8 minutes of lying really, really still to get that image. We don't know anything about activity, just kind of structure from that. But those functional images I showed you are, pretty good for how long they take. So most recently, using some really new advances in fMRI technology, we can get our functional images down as small as 2.5 millimeters by 2.5 millimeters by 2.5 millimeters. So like, that big chunks of brain. And that takes about 1 second to get a full 3D brain image to get one of those. And of course, when you do a task, you would be recording one of those every second for several minutes on end, and then like looking at the aggregate activity across a long time span for various cognitive events. Okay, but that's just sort of like the detail level we're looking at. Some sort of practical things. If you want to buy one of these just for your own home use, it's going to cost you around $3 million, give or take $1 million or $2 million for like, if you want the bells and whistles and all this stuff. Plus, it's going to cost you around $100,000 to $200,000 a year for basically the MRI tech to come around and service your machine, because they break down a lot. So that breaks down to, if you want to rent one, if you'd rather rent than buy, that'll cost you about $480 an hour right now at Yale, and that's sort of how they have to charge in order to like make up the overall cost of the machine. So it's pretty expensive, but that is still cheap, like we said, compared to PET or SPECT, which is like $2,000 for like an hour-long scan of a subject. A few other facts and figures. So those of you that had the MRI before, it's pretty noisy, right? Yes? Kind of annoying. So, to protect people's ears, that's from all of the moving parts that actually go into like adjusting the magnetic field at every moment. So for that, people have to wear protective earplugs and headphones. It also limits what you can do in terms of how you test people's, auditory processes, right? Because like you can't eliminate the sound, or else the MRI is not on. So that's a bit of a bummer. One other thing is that to get a magnetic field that strong, it requires really high levels of electricity and really cold, superconducting materials. So you have to have a big store of liquid helium and liquid nitrogen nearby to keep, obviously the person in the MRI machine is kept at room temperature, but the stuff supercooling the magnet is at minus 269 degrees or so Celsius. Apparently there's actually a helium shortage in the world these days. I don't know if you guys have heard about this, but we're running out of helium, so it's getting really, really expensive these days. Say goodbye to your helium balloons and your high voice helium huffing parties, because it's going to get very expensive very soon. Hopefully we'll find more helium somewhere. But yeah, so it requires supercooling. That gets expensive, and if you run out of that stuff and your magnet heats up and ceases to become a magnet because it's no longer superconducting, that's a really expensive service visit. That's like, I think it's like $50,000 or $60,000 to get your magnet running again. So the magnet can never be turned off, basically, unless you're turning it off forever to like get a new one or something. So because it's really, really, really expensive to get it running again. So because of that, we have very strict safety requirements, because the magnet's always on and it's very strong, right? So like we said, you don't want to go in there with your keys. You don't want to go in there with your credit cards. If you've had an MRI before, they've told you all this stuff. You definitely don't want to go in there with any of your private body piercings on if they are magnetic, because that could get uncomfortable. You don't want anything getting sucked into the magnet, and hurting the person in there. So we don't have time to watch all of this, although it is a gem. But I thought I'd show you an excerpt of the safety video they make us watch to illustrate how important it is not to take metal into the MRI machine. This has the, so sorry about the video quality because it's from like a super old video. Um, this is from GE, who makes MRI machines. So this is from like the 80s or something. So I'm going to skip through a lot of it. The unfortunate side effect that this has is it's supposed to tell you like how awful it is to do all this stuff. And like many safety warnings, what it really makes you want to do is totally do all this stuff. But yeah, so there's the whole thing about the all the stuff I've already told you about the car magnets and stuff. A little bit about how MRI works, how the magnets are. All right. So here they've got some dudes who look. So they've got like in their research facility, they've got like an MRI system set up.
Its distance from the magnet and how it's oriented to the field
You
line.
got these guys
What
who
will be
look
the
like they're
force
having a lot of
exerted on a
fun
2-pound
doing
pipe
this.
wrench?
So this is like if you took an ordinary wrench into the
Securing
MRI
one
magnet,
end of a line to the wrench and the other to a strain gauge and
so you
winch
see it's not even
allows
at
us
that
to
distance
measure
from
both the force and distance
the actual
from the
MRI
magnet
to,
bore. At a
hover it
distance
in
of
midair.
2.5 feet, the gauge reads 3.7 pounds.
So that's
Reduce that to 2.
That's pretty scary. So then they're going to like watch it get stronger and stronger. Plot the force, but plotting the force is not that interesting. Let's see what happens when you just like toss some stuff into there.
Into the magnet.
So this is
A radar gun
I
will
went a little too
indicate
far.
the
So that's a sorry. That's like a little tennis ball filled with steel shot, I believe. So let's get back to that. All right, so he'll tell you.
For this experiment, we'll take a rubber ball filled with steel shot and toss it into the magnet. A radar gun will indicate the speed.
That's it. Clearly having a little bit
In
more
these
fun than
tests, we measured speeds of
probably
20 miles per hour or more. But more important is the rapid acceleration and unpredictable direction. The ball can take any number of routes as it flies into the magnet bore.
So imagine if you
Imagine
were,
the damage
that's
that
probably
could
weighs a
happen
couple
if this were a
pounds.
wrench.
If you were the person in there, that would not feel good if that were to hit you. And there have been, like this should not scare you from getting an MRI if you have to for medical reasons or research reasons, but there have been some pretty severe accidents with people accidentally
Have
taking.
you ever had anyone go in who forgot about some of their metal?
Well,
These demonst-
yes. So, I have all kinds of stories. Thank you for asking.
That was not set up. I
Yeah.
actually did not.
Let me see if I can pause this without. All right. So, a couple of stories, of varying levels. So, actually, when I first interviewed for my first research job, I had borrowed someone's car to go for that interview, and I was not used to having car keys in my pocket because I didn't have a car at the time. So I forgot to take that out, and so like my pants pocket just kind of went like when they walked me in to tour the magnet. So that was a little embarrassing. Fortunately, like it was not a big deal. By the way, you might have noticed like now because I do this kind of research, I always compulsively take everything out of my pockets when I walk into a new room because it's just what you do every time you walk into the MRI area. So one time the person who was actually one of the techs, you have a person whose job it is to like run the scanner. One of the techs who was supposedly also the person in charge of safety for our research center, this was at not Yale, this was at the place I worked before, took a metal tape measure into the scanner area. I think she did it just to see how hard it really would pull. Now, fortunately, there was nobody in the MRI at that time. But like she got. The problem is like if you guys remember how magnetism works, it's an inverse square law like gravity. So it means that if you go twice as far out, the field is 1/4 as strong. But that means if you decrease the distance between you and the magnet by 1/2, it gets 4 times stronger, right? Instead of just twice as strong. So it's like you can be like, oh, this is kind of pulling a little. Oh, this is kind of pulling a little bit more, and then all of a sudden it's pulling so hard you can't hold it. So that's what happened, and it got like sucked into the scanner and stuck to the side, and like that ball did, and like it was stuck there so hard that she could not pull it out by herself. So we had to get like 3 people like kind of pulling on each other and the tape measure to like pull that out. So that was not good. Those are the main ones. I mean, I will also tell you that like I have been known like when I was working in the magnet in that same research facility, like I was setting some stuff up that came with instructions in like a binder, and I have been known to like use the MRI to like stick the 3-ring binder to the side of the magnet so it would stay open. I don't recommend that, but it was very useful. But that was like a very small amount of metal. Yeah. Braces are usually okay because they're usually okay safety-wise because they are made from metal that does not get sucked into the magnet. So, like we said, only really iron/steel, which is made from iron, and like magnesium and nickel are the only ones that get really attracted to the magnet. Now, other metals, the problem is not so much for safety, although that's also, like, they may twist. So, like, I've gone in just for a quick test with my belt on before, and, like, you can feel your belt buckle kind of twist around in the magnetic field, although it doesn't, like, suck you in. But the other problem, and I have a story about this too, is that metal acts as an antenna. So the problem is not necessarily for safety with other types of, as it is with like iron, but the whole idea of this MRI is that you send out a radio pulse and then like measure the echo back. So if you've got another antenna kind of soaking up those radio waves, basically what happens is you don't get a signal in that area because the metal is absorbing all of those radio waves. And so we had a person go in with like a little ponytail holder that, you know, the ones with the little metal clip. So she went in with that, and we didn't see that. We thought it was just a pure elastic ponytail holder. And then we ran her first scan, and it looked like she had like no back of her head at all, which was like a little disconcerting until we figured out that that was why. So like that's a bigger problem, is that if you have metal in you, like fillings are usually okay because it's not very much, but if you have a lot of metal in you, it can just mess up the signal, and you just can't get anything useful out of it. Yeah. Any other questions before I show you more videos of things you should never do? Keep hitting the spacebar, but that advances the slide. All right, so just a couple more like demonstrations of how strong the magnet is. So they're going to set up this like little wall thing that's like a ceramic.
Never be attempted by anyone outside of this controlled environment. Show the overwhelming power of the MR system magnet and the sudden violence that can result from carelessness or lack of knowledge.
The guy just tosses the wrench kind of lightly towards the magnet. So yeah, don't want that happening if that was your head. And then they go on, let's try it with a metal pail. So the metal pail will, of course, also stick to the magnet. Okay, that doesn't look good. And then for good measure, let's set up a few bricks. Okay. So yeah, the basic moral of that story is do not take your big heavy pipe wrench into, never do that, is the moral of that story. Okay. Yeah, so basically safety is important, kids. Don't try this at home. And they are really serious about safety these days. You know, there have been a few accidents in hospitals, but overall it's very, very safe. Safer than driving or any of that other stuff. Okay, so just a quick summary of the fMRI method, then we'll talk about a little research, you know, with whatever time we have. So, you know, it's got okay temporal resolution, a couple of, well, you know, we can scan every second or two, get a scan every second or two, but of course your stupid brain is limited in how fast its blood responds. But, you know, it's got okay temporal resolution, better than PET or SPECT. It's got pretty good spatial resolution, you know, a couple of millimeters. It's reasonably convenient. Most people can do MRI. You know, you can't do it if you've got a lot of metal in you or something, but it's, you know, fairly safe, no radiation, whatever. Disadvantages are that it is expensive, although not as bad as some things. You do have to worry about metal and safety issues some, and the temporal resolution is nowhere as good as some other things. So there's that. So now I think we can do a little bit of talking about like some actual research. Hopefully this will make some sense to you guys. If not, like please feel free to like let me know what doesn't make sense, because you know you get into the jargon of explaining this to people that like know a lot of the jargon, and you know I always forget like what people study when. So, but so to like contextualize like the mental, is that a question or are you just stretching? Okay, cool. One of the mental processes that like our research group is interested in is attention, which I don't know if you guys are, you know, we all know what it means to like pay attention, but as a relatively formal definition in psychology, although it's pretty much like the informal definition, right? It's like, well, actually, William James, our master of psychology, even back in the 1800s, defined it really well. He's like, everyone knows what attention is. It's the stuff that you pay when you pay attention, right? So it's taking possession by the mind in clear and vivid form of one out of what seem several simultaneously possible objects or trains of thought. It implies withdrawal from some things in order to deal effectively with others, right? So like, you know, when you're looking around the room, there's all kinds of stuff you could be looking at or paying attention to, right? But we know from, and actually you probably feel like when you're looking at, like when I'm looking at all of you guys, I feel like I see all of you. But we know from research that actually you're only really able to pay attention to a very, very, very small amount of the perceptual world. So really, like most of what's in your visual field, you like never notice what's going on. So, you know, if I'm looking kind of straight ahead and one of you sticks your hands out and I'm not like continually scanning and changing where my attention goes, it's a very good chance that even if you're right in front of me, I won't see your hand up, for example. So we actually like don't have as much attentional capacity as we think, but that's a story for another time, probably. So we generally think about attention when we talk about vision, right? Because it's very easy to see that. So if you have never done one of these types of tests before, I've got a simple attention style demo to kind of show you the difference that attention makes. Hopefully this will work on you guys. So what I'm going to have you guys do is keep your eyes on this little cross here. We call it a fixation cross. So try to fix your eyes right there. Don't move your eyes. So pretend we're out at the club and you see somebody who's like really super hot, and they're right here. But you don't want to like make direct eye contact with them because that's like a little too obvious, right? So keep your eyes here. You're going to kind of be paying attention out of your peripheral vision to this place right here, right? So keep your eyes here, but pay attention to what's here. So I'm going to flash something super fast here, and we're going to see if you can detect it, okay? So keep your eyes on the cross. You ready? Here we go. All right. So what did you guys see out here? So what about on the left side where you were supposed to be paying attention? Saw. Okay, so some sort of building, right? Now how about over here where you were supposedly not paying attention? Okay, as a face. Now let's see how this sometimes works and it sometimes doesn't, depending on how good you guys were paying attention and how fast this display is. Let's, by show of hands, was the person, so this was a face over here, was the person's face, first of all, was it a man or a woman? Raise your hands for man. Okay, now raise your hands for woman. Okay. Now let's ask three things. So now, was that person a younger person, like in their 20s, or an older person, like in 60s, 70s? Like younger or older? So hands for younger. Okay, hands for older. And just take your best guess if you're not sure. Okay, so most people think male and younger. And lastly, let's do race. So was the person white or African American? Raise your hands for white, and raise your hands for African American. Okay. All right. So most people said male, young, and white. Although there was some disagreement, I think, on all three. Okay. So we're gonna do it again. Now, don't pay attention to that same location. Actually, I'm just gonna do it a bit slower, but still try to keep your attention focused out here. Okay. I'm gonna do it, but like don't cheat and pay attention out here. Pay attention. See if you can get more detail about the building out here, okay? It's just going to go a bit slower, okay? So keep your eyes focused here. Pay attention to see if you can get more detail about this building, okay? You ready? Here we go. Okay, now some of you may have cheated. I'm sure some of you did. But did anyone change their mind? So did you get more detail on the building that time? You could see like a little bit more what it was. Okay, good.
There was no way that was the same picture. Are you kidding? Was that the same picture?
Well, so did anyone change their opinion of what the person's face was?
Yeah.
Okay, so people changed their opinions. So now I'll just show you. It was in fact Justin. No, that's a lie. It was not Justin Bieber. It was actually this lady here. So it was the same picture all of those times. Yes, it was not the beads. It was this lady. But so you guys, most of you were correct that it was a young person, but a lot of you got the gender and the race totally wrong. And you'd think that, gender and race are pretty easy to pick out of a face, right? But that shows how much different. So it shows a lot of you were very good at paying attention over here to this like church or city hall or whatever that is. So if you were fully paying attention over here, it's true. Not much got in of the face over here, right? So that's attention at work. That shows you that something can be like right there in front of your eyes, and you don't get a lot of detail if you're not paying attention to it. Okay, so that's kind of cool, but it's not exactly what we study. So what we study is a different type of attention. So you probably know intuitively that you can pay attention to stuff going on inside your own head as well as stuff out there in the world, right? So like if you drive, oftentimes you'll find that like you start thinking about stuff in your own head and like, you drive for 50 miles and all of a sudden realize that you missed your exit. Well, I did not do that today, thank goodness. But, or you're like walking along thinking about lunch or what some friend of yours said or something. You like walk into a tree branch or whatever. Something that's like right in front of you. You have no reason to not see, but you don't see it because your attention's all pointed inside, not to what's coming in your eyes, right? So just to highlight, back from this William James quote, just to highlight one little bit of it, attention can also be directed to trains of thought as well as to like visual items or other senses. So a question that we have in our field of research is, to what extent or does attention within thought work similarly to how it works, among things you're perceiving, or in what ways are these types of attention similar, and what ways are they different? So here's one study that we did with fMRI to kind of get at this question. So a little bit more background. We briefly, I think, alluded to like the fusiform face area, which is an area of the brain that activates mostly specifically for faces. And there's also an area called the parahippocampal place area that activates mostly specifically for looking at pictures of places. And these aren't, again, we're not entirely sure exactly what these areas do, but we do know that this is somehow involved in face perception. This is somehow involved in perceptions of visual scenes. And there are various other visual areas that are like this. There's one that activates for like objects, one that activates for bodies, but not faces, and a few others. But we know that we think of these as visual areas mostly. We think they're involved in visual processing. But one hypothesis we have about how your memory works when you're not actually seeing something, one idea is that when you're trying to remember something that you have seen, one way we think that might work is that these aren't totally different brain areas involved in these processes. We think that you might reactivate the patterns of activity that happened in these perceptual brain regions when you're trying to remember them. So the idea is that you kind of get a faint like echo or a faint replay of the original visual activity when you try to remember something that you perceive visually, right? Because I don't know if that makes sense intuitively, but hopefully it does, that that's an efficient way to use your resources, that you kind of do a replay of the same activity, and that, if you have I don't know how you guys are at mental imagery or imagining things that you've seen or heard, but if you're really good at imagery, some people say that they can you can almost see the thing you're imagining. Does that kind of make sense to people? Okay. Oh, I should have replicated this. I can't not tell you this study because it's so cool, even though it has nothing to do with neuroscience. Can I tell you this? Somebody put a blank screen up here. Can't get past that one. Okay. I thought I had a totally black screen, but maybe I didn't. All right. Well, if I show you a mostly black screen, this is one of the greatest psych studies of all time. This was done in the 20s before we had any kind of neuroscience. I'm going to ask you guys, look at this blank screen and just imagine a banana on the screen, okay? Like a regular, standard banana, okay? Now, everyone picture it in your mind as clearly as you can. Everybody good with that? Okay. Now, was the banana you pictured, was it sitting like this, kind of like that way, or was it standing up on end? So raise your hand if it was kind of lying down. Okay, now raise your hand if it was standing up. Okay, so about even. I think a few more lying down. Okay, so different angles of banana. Fair enough. That's no judgment. So I think most people a few more people tend to imagine it this way than this way, or other ways. But there was this really cool study in the 1920s where what they did is they did exactly what I just told you to do, but they extremely faintly projected a picture of an actual banana on the screen. So faintly that you couldn't tell that you were looking at anything but a black screen. And I didn't do this here because I don't prepare that well for that kind of thing. But they put an extremely faint transparent banana on the screen with a film projector, and even though people had no idea that they were seeing a very light impression of a banana, it affected the orientation of the banana they imagined. So if you projected the banana upright, almost everyone reported the banana they imagined as being upright. And if they did it lying down, more people said it was lying down. So all that is to say that we think that your perception and your inward mental ability, your memory and so forth, interact a lot more than you probably realize, even when you're not aware of it, right? So your thoughts probably affect your perceptions more than you realize, and your perceptions probably affect your thoughts more than you realize. Yeah, if you could figure out you'd have to figure out a way to do it. I guess you could have people imagine a song or something like that, and have it playing very, very faintly. But yeah, it should work. In theory, it should work the same way. There are so many more studies on vision than there are on hearing, just because it's much easier to run them usually. But yeah, should. Okay, so the basic idea though is we want to kind of see if we can get some kind of measurement that shows that we replay brain activity from vision during like mental imagination. So in other words, what we want to see is if I show you a face and like a picture of a scene or a place, and I have you think about one or try to imagine one, will we actually see activity in these visual brain areas that reflects the category of the thing that you're seeing? Does that basic idea make sense to people? So if you think of a face, will your face area activate? If you think of a place, will your place area activate? Even if you're not actually seeing either one, right? Is that everybody cool on that? Okay, so this is what we actually did. We did a study with a few conditions. It's not clear. These are kind of faded out, but it doesn't matter. So we had 3 conditions in the study. So basically what would happen is people, on each trial, and of course you do a lot of trials of this, but on each one of the trial, people would do one of 3 things. So in one condition, which we called the refresh condition, because you're kind of refreshing your memory, people first saw just a cross on the screen that just let them know the task was about to begin. Then they would see 2 pictures. In this case, I reused that picture here. It was always one scene and one face. And then there's a brief blank period for 500 milliseconds, and then they'd see a little arrow on screen for a second and a half. And that arrow basically meant imagine or think back to or visualize whichever item the arrow pointed to. So the arrow points to the right, so people try to mentally visualize that face just for a second or 2, not for very long. So we contrasted that with a condition where instead we use the same stimuli on the screen, but we put the arrow at the front. So what this basically meant is the arrow points to the right in the beginning. That means when the 2 pictures come up, do what you did with that kind of Justin Bieber task earlier and only pay attention to the one on the right. Just ignore whatever's on the left side of the screen. And then here the cross shows up at the end just it doesn't mean anything. Okay? So here we're having people pay attention in memory to one of the 2 items. Here we're having them pay attention in actual vision to one of the 2 items. In both cases, though, people are visually presented with one of each, face and scene, but biasing their attention one way or the other. And then in this third condition, which we called ACT, because people take a physical action, we show the fixation cross, show the 2 pictures, but then at the end we just show a gray square, and what that told people to do was just press a button with your finger that they were holding a little button box in their hands. And that's just basically like a replacement for something to go here. What that basically meant is what we've got in theory in each of these trials is they've seen a face and a scene, but they haven't paid special attention to either one, right? So in theory, what we should have is a set of conditions where people paid either particular mental attention to either the scene or the face. They paid sort of eyeball attention, perceptual attention to the face or the scene, or they looked at them both on average about equally and didn't pay special attention to either one and just pressed a button. Does that kind of make sense to people? So the amount of actual visual things that came onto their eyeballs were the same in all these conditions, but different amounts of attention, and the attention was directed either while the things were actually on screen in this case, or after they disappeared in this case, right? So the question is, what happens in these visual areas when you do that? So let's look just at, so this is an image of where your parahippocampal place area is. In your brain, this is a coronal section, so this is slicing it this way. So it's kind of towards the back, if I took the slice from kind of back here-ish, I think, kind of towards the bottom, just above the cerebellum. Did you guys do a cerebellum stuff today? So that's the cerebellum there. It's kind of blurry. And just above the cerebellum, that's about where your PPAs are. Near your hippocampus, which is why it's called parahippocampal. That's near where HM, the amnesia guy, that's near the area that was cut out of him. So what we're going to look at is, so these blobs are just to show you where the area is that we're looking at. So now what we're going to look at is the amount of fMRI signal associated with each of these trial types. So we're ignoring all the statistical analyses and all the boring junk that goes into getting these numbers. This is just looking at an index of how much these place areas activate for each of those conditions, okay? Is that cool with people? So it's just going to be a bar graph, and this axis just basically means the amount of activity. So this is the amount of activity we saw in that area for the ACT condition, which is kind of the baseline condition. What that means is like, we're going to see, and these units are like meaningless. They don't mean like a quantity really. They're a relative amount. But that's how much was going on in this brain area when people just saw a face in a scene and pressed a button. So, obviously from just seeing a scene, you're going to get a certain amount of activity in this scene area, right? Now, if we tell people to pay special attention to the scene, you get a little bit more activity. So that's cool, right? Well, I mean, depends on your definition of cool, but that's what we would expect, right? So if you pay a little bit more attention to the scene, a little bit less attention to the face, you get a little bit more activity in the scene area. And if I tell you to attend to the face and ignore the scene, while you're actually seeing them, you get less activity in your scene area, right? So again, that sort of makes sense. Now the question is, what happens when we tell you just to think about one or the other? And the answer is, it looks almost exactly the same as if you're directing your attention while you're actually perceiving things. So you get a little boost of activity when you think back to the scene, even though it's not on screen anymore. And you get an actual suppression of activity when you divert your attention to the face even after they're off screen. So this is actually, we won't talk much more about this, but this is actually the coolest part of this study is not only does thinking about the scene after it's already gone off the screen, not only does that make your place or your scene area activate a little bit more, but if you think about something else, it actually kind of cuts off activity in that brain area, even relative to if you thought about nothing at all, right? So it actually kind of like tamps down activity in that area when you think about something that's competing versus if you think about nothing in particular. Does that kind of make sense to people? That makes sense to people? Yeah, question?
Have you ever tried making someone look at a face and think of a scene?
Not exactly. I mean, these kinds of things get tricky because, I'm glossing over a lot of details. Like, so when I say the face area prefers faces, the dirty secret is like the face area also shows some activity to scenes, to objects, to other stuff. It just shows the most activity to faces, right? So like, usually when we see activity there, it's associated most with faces. But like, it gets tricky then if you talk about stuff like that, because if you are showing one thing and thinking of another, and you just have an overall readout of activity, it's hard to tell like what of that activity is coming from what you're actually seeing and what's coming from what you're thinking. But people have done studies, so like some of the early studies on this area that led up to this, like people have done studies where they kind of do transparent overlays of a face in a scene. So it's like 50% one, 50% the other. And then you can manipulate like which one people pay more attention to. So tell them to like try to, on this kind of weird like transparent image, try to focus more on the scene or the face. And you can kind of push around activity in these areas that way, yeah. But that's more, your visual attention more so than your thought. The thought stuff gets tricky. This is more of a philosophical issue or more of a logistic issue you guys don't have to be worried about. But once we've determined that just thinking about stuff can affect activity in areas of the brain that we think of as visual areas, how do we know what's a visual area, right? Because like if you can just generate activity in these areas by thinking without seeing anything, then what does it mean for it to be visual, right? Like this gets really tricky, right? Because even as it turns out, like even really early areas in your visual processing stream, or like areas that are thought to be purely driven by visual elements, can be affected by like what you're thinking about. So not as strongly as like what I'm actually showing you visually, but still it gives, you can't really just call it a visual area anymore, right? It's kind of a visual area and thinking about visual type stuff area, but then like what does that mean, right? So yeah, once you learn that thinking can affect sensory areas, your whole worldview sort of collapses a little bit. But let's not worry about that too much right now. Any other questions so far? Okay, so that's like a sort of basic idea. Oh, so this is maybe another way to visualize the same thing. So if we actually look at activity in one of these PPA areas, so again, this is your scene selective brain area. It looks basically the same in each of these. I just show like you've got one on your left, one on your right side of your brain. That previous figure was average, but here you can kind of see them separately if you want to. This is instead plotting, if you guys remember that demo I showed you of the fMRI activity over time, this is showing you that kind of plot where 0 is the point where this trial began. And this shows about 14 seconds of fMRI activity in this area. And you can actually, what's kind of cool about these plots is you can actually see kind of the trial unfold in slightly, again, it's slightly delayed because your blood flow is slow, but you can kind of see the trial unfolding in real time. So the gray plot here is that ACT condition, which is our baseline. So you see what happens is at the beginning of the trial, the activity is basically 0. It kind of raises up a little bit, reaches a peak about, 6 or 7 seconds in, which makes sense because you saw the actual visual face and scene about 2 seconds into the trial, and then about 5 seconds after that, you'll get your peak activity. So about 7 seconds in, you've got a peak. Actually, I think it's 8, but whatever. And then it kind of like dips back down and goes back below baseline, and then we don't really care what happens after that, right? So that's what happens on a typical trial where you don't pay any attention. Now, if we look at the red lines, dotted and undotted, that's when I tell you to pay visual attention to either the scene or the face, right? And what you see is all the lines kind of start out together up to about here, and then you see here's the gray line, it kind of goes like that, but the red line shoots up a little faster and kind of reaches a peak earlier and kind of stays a little bit higher throughout the rest of the trial, whereas the dotted red line, where you're paying attention to the face, never shoots up quite as much, kind of tails off earlier and then dips back down, right? So basically what you can see is at the point in the trial where the items came on screen, people chose to pay more or less attention to one or the other and had more or less activity in one area. And the coolest part, I think, is that if you look at then the blue lines, which is where you're thinking of either the face or the scene, those blue lines go right along with the gray line, right, the gray baseline, up till this point. So a little bit later on, and then you get that arrow cue after the actual face and scene are off screen, and at that point you see the blue line shoot up a little bit more, even above the gray line, and the dotted blue line just takes a nosedive, right? Because that's where you know that you're just cutting off thinking about that scene entirely and focusing on the face, right? So you can actually kind of see, now it's super delayed and very blurry in time, right? It's not very precise in time, but you can actually see the progress of your sort of brain's decision-making process unfold in the scene selective area over the course of that trial, just shifted a few seconds. Does that kind of make sense to people, how that plays out? So that's kind of neat. This is actually, I've never seen data this beautiful in any study I've ever run since. Like, this is actually really clean looking data for fMRI. And of course, this is averaged over many trials and about 15 participants, I think. So this is not like a single trial or anything, but this is pretty clean data, even for averaged over a bunch of people. Okay, so just to show you another couple things from this same study, this is probably the sort of brain map you may have seen in like the news before. So we did make one of those. This was to ask a separate question, a slightly less important question, but still kind of interesting one. If we forget about like whether people are paying attention to the face or the scene, and just instead ask, regardless of face or scene, are you directing like sort of eyeball perceptual attention, or are you directing mental attention within your memory? So the red areas are areas that are activated more for paying visual attention than that ACT baseline condition, right? So that's, we're not paying attention to anything at all. That's just sort of our baseline. So the red areas are where paying visual attention activates them more than that. The blue areas are where paying mental or memory-based attention activates them more than the baseline condition. And then the purple are the overlap, right? So what you basically can see from this is in this map, there's a lot of blue areas, a lot of red areas, and a fair number of overlaps, right? And a fair number of areas where like here is a great one, where you've got some overlap, but then kind of the clusters of activity shoot off in different directions. And like what these actual brain areas do is probably not that important for you guys or what we think they do. But the kind of take-home point here, I think, is that these processes, this sort of supports our idea that these processes of outwardly directed attention and inwardly directed attention are partially overlapping and partially not, both in terms of like overall areas that they recruit. So there's some entire areas that are all blue or all red, but other areas that like part of that region is used in both processes and then part of that region is used in just one. Is that a question? Or just a stretch. All right, any questions? So just the last thing I'll show you from this study is the same thing, but in a much cooler way, I think. So this is just rotating these blobs of brain activity in 3D. It's the same blobs you just saw on the previous slide. So red is for paying visual attention. Blue is for paying internally directed. The blue doesn't show up that distinct from the purple in this, sorry. And then the purple is the overlap of both. So that just lets you visualize it in 3D. I hope you enjoy that because that took like a week of my graduate school to figure out how to make this type of. And actually, I've completely lost the code that I used to make this figure. So that's the only one I'll probably ever make. But enjoy it, feast your eyes. I whip it out as often as I can because I don't want that time to go to waste. Okay. So basically, what we concluded from this study is that turning your mental attention to something does activate visual areas that are related to that, whatever type of item you're thinking about, which is very similar to effects we see for perceptual attention or visual attention, and that there's a lot of overlap in areas associated with these things. Okay, any questions about that study so far? How do we want to take more breaks, or when do we wrap up?
All right, let's get our attention back. And before we get back to the slides, I was wondering if people had questions just for Matt about being a scientist or any kind of general questions.
How do you become a researcher like that?
Do like in cognitive neuroscience or just general? Well, you make a series of bad life decisions. No, I'm just kidding. It's a wonderful life. Not at all frustrating ever. No, I mean, basically, like, my actual background, like, I double majored in college. I was a cognitive science and English double major. And then I, at least in my case, was like, I don't know what I want to do after graduation. So I took a job as a research assistant because that was one thing my cognitive science degree qualified me for at that neuroimaging research center. And then, kind of spiraled from there. Then I decided to apply to grad school. Once I had the PhD, I kind of like did a couple of academic jobs and doing my postdoctoral job and so forth. So like, and then the hope is that you go on to be faculty at some university, and that's the dream.
Or a high school teacher.
Or
that's way
better.
Or
I mean, whatever you want to do. But if you're wanting to like do daily like research in this stuff, that's sort of the standard path. But there's also private companies and other educational, paths you can take. But anyway, usually like. You usually major in it in college. I mean, you probably did some research while you were in college.
Do research
yeah, I didn't do a lot, but most people do.
Or after college, and then think about doing grad school.
You
do not want to not have any research experience really and apply to grad school. That is a terrible idea.
Yeah. You want to know if you like the lifestyle before you get into it.
It is very challenging. It will test you on levels that you have no control over. Like if you study hard for a test, you will typically do well on that test. Research is so beyond your control.
Well, the whole reason you're doing it is because you don't know what the answer will be. And if the answer is not what you wanted it to be, then you just wasted, a couple months of your life trying to find out that the answer is nothing interesting, right? But like
nothing.
There is no answer. But that's a lot of like, it's a lot of like trial and error and figuring stuff out and like, taking the long view of things. But some people really dig that and some people get frustrated and so forth.
While doing this as well?
Yeah, if you're really crazy, you can do an MD PhD. I shouldn't say crazy. Like, yes, you can do an MD PhD if you want to be in school forever.
10 years.
Yeah, I mean, it's just a PhD is 5 or 6 years. MD is, I guess, what, like 3 years plus a residency and an internship and all that stuff. So yeah, you're in school for a long time, but like, that's something people do. It sort of depends on whether. What you're most interested in, whether that helps you. Like, for what I do would not really help me because I don't do anything medical in my research. But like, if you were interested in like, a psychiatric illness and wanted to be a psychiatrist / researcher, or epilepsy and wanted to be a neurologist / researcher, that might be something you would want to do. But it is a lot of school. However, on the other hand, the PhD often subsidizes your med school, which, if cost is an issue. I don't know if that's always the case, but definitely at Yale it's the case. Like,
I think it is.
You don't have to pay for your med school then, which is kind of nice.
And you don't pay to go to grad school for most cases. It's for science.
You get paid to go to grad school.
Things.
To get to do a PhD, I should say.
To do a PhD,
not a
they
master's.
pay you.
You don't get paid a lot.
Yeah, not a lot.
A lot of fancy meals.
I actually, this year, like in September when I came back here, is the first time I've made more than I made when I was like 24 and just had decided to go to grad school. So like it went down from what I was getting paid as a pre-grad school research assistant. And then it like gradually went up a little bit, but only now am I back to more than when I was like 9 years ago. Yeah. Yeah. And it's still not much.
Just, we're going to talk a little bit about EEG, because I think it's important for you to know. But then we'll be done with A. Watch out. Yikes. You okay?
Yeah, I'm fine.
We all good?
Sure?
Okay.
Okay. So, yeah, hopefully. So I know you guys are probably, your heads are now full of information about your heads. And hopefully you had that extra % of space in your brain and you did not have to throw out like how to write or do math or something to learn this stuff. But a little bit more. So like Sarah said, we're going to talk about EEG some. EEG is the oldest thing that we can kind of call neuroimaging. Like I said earlier, we first did this in humans in 1924. I mean, if you want to get technical, it's not really imaging because we don't get a picture of the brain exactly, but you are recording from the brain, from all over the brain. If you guys remember how we said TDCS worked, this is sort of the opposite idea from TDCS. So in TDCS, we said we'd kind of send some electricity through the skull and the scalp and make it affect your neurons. The opposite thing also happens just when your brain activates. So when groups of neurons in your brain are active, that affects, the voltage in the space around the neurons, which then, kind of spreads to your skull, which kind of spreads to your scalp, and which can be very, very, very faintly detected by electrodes on your scalp. So as you might expect, it's not a big signal to begin with. We're talking about like millivolts of voltage in your actual neurons and the space around your neurons. And by the time it kind of makes the journey through your skull and your scalp and so forth, we're really talking about microvolts of activity. But with a sufficiently good set of electrodes and a good amplifier, you can actually pick it up and make some sense out of it. So just to get the terminology straight, so EEG stands for electroencephalography, electro for electricity, encephal for your encephalon, your brain, and ography, like writing, like anything ography, like geography. It's a very similar idea to a few other ographies, which I hear you guys are going to be doing one of, would you say tomorrow, next day? Tomorrow? So you won't be doing electrocardiography, probably not. But if you've ever had an EKG or an ECG, hopefully you have not. But if you ever have heart troubles, and they put the electrodes around your heart, it's a very similar idea. You will be doing EMG, electromyography, so that's recording electrical signals from the muscles. There's also electrooculography, which is recording signals not from your eyes, you're not sticking the electrodes in your eyes, but from around your eyes. So your eyes actually create very large voltages when they move, and you can record that by putting electrodes like here and here. And we actually do that when we do EEG, we actually also do EOG, because when you move your eyes or blink, it can create noise in the EEG signal. So we actually also track your eye movements so that we can make sure you're keeping your eyes still. So all of these are basically the same idea, which is that something in your body generates an electric current or a voltage. So you put an electrode on it or near it, and you measure the voltages it creates, right? So with your heart, you put the electrodes over your heart. With your muscles, you can, attach it either to your muscle tissue directly or over your muscles and so forth, right? Okay, so all the same basic idea. You put a wire on something, and if it's got an electrical current, that current goes up the wire. Pretty basic idea in principle. And then, of course, as usual, there are tons of small details to worry about. So with EEG, the electrodes go on your scalp. And what essentially you get in each electrode is essentially a summation of all the activity of a lot of areas underneath each electrode, which are kind of heavily blurred over space, meaning that we have very little spatial precision. So the electrode itself is about the size of a dime, usually. So that's how big the electrode is, but the area of the brain that could be representing activity from could be very big, could be several centimeters. We're not actually even, the math and the physics of how electrical signals propagate through the brain is actually really complicated. So we have very little spatial precision. And like we said, it's very small electrical signals, a few microvolts. So basically our spatial resolution is terrible, but you can get really good temporal resolution. So you may not know exactly where something is happening, but you have a pretty good idea of when it's happening. Usually on the order of 1 to 4 milliseconds. It really just depends on how fast you want to acquire the data, but 1 millisecond is pretty good. So this is one of our undergraduate research assistants who's doing summer research with us this summer. So she volunteered to be my subject in something just recently. That's her wearing the EEG cap. So these electrodes are usually nowadays they can just be naked electrodes that you just kind of stick with a sticker onto someone, but it's a lot easier when you're recording. We record from 64 electrodes scattered all over the head. So the easiest way to do that is to put them all in this kind of swim cap thing, and then that kind of puts them all in standardized positions on your head. So that's her with the electrode cap on. That's this one stuck to her face. These four you'll see, those are actually the electrooculography, the EOG electrodes to measure her eye movements. So those will measure voltages when she moves her eyes up and down or left and right. So that's what that looks like. That's me. I was also a subject in my own study, so that's just to show that I will also volunteer for my own stuff. It's sort of a pain in the neck. You have to put this gel in the electrodes. I'll show you that later, which is kind of sticky and gross. But that's kind of what it looks like to do an EEG experiment. And Sarah knows quite a lot about this because
I've been in almost all of math studies during grad school.
yeah, I forgot to mention that, but she was one of the brains represented in those other studies, and is going to be one of the brains represented in what I show you next. Yeah, and has stuck with me through some tough studies.
Tough times.
Yeah. Yeah, so there's a few other related things that I'll just zip through quickly. There's intracranial EEG, which is a similar idea, except you put the electrodes directly on the surface of the brain. Again, usually this is something you only do for like epilepsy patients before surgery. You don't usually crack someone's head open just to put an electrode array on them. But if their head has already been cracked open for medical reasons, you may as well do some research while they're in there. Because they're basically just like, usually they're kind of like sitting around the hospital waiting for seizures to hit so that the doctors know where the seizures are coming from. So like they're happy to do experiments for you. Well, somewhat happy. They'd be happier, I'm sure, not to be in the hospital in the first place. But yeah, so you can do the same thing, and you get a little bit more spatial detail that way because you're not having to go through the skull. I have some pictures of this. If you don't like looking at brains during surgery, now's a good time to close your eyes. It's not like super gross, but here we go. Okay, yeah, so warning, here's your warning, close your eyes. So that's you don't see too much. It's like, that's a better one probably. So that's someone with their head cracked open. You see that it's not the best with the brightness, but you see there's this little like array of kind of, it looks like a bunch of little buttons in kind of saran wrap that you kind of stick down to the surface of the brain. Okay, and so those you get like about a centimeter apart. And the nice thing is that then, because the signal's not going through the skull, you're recording what's directly under that electrode. So it's not as precise as if you're recording from a single cell, but it's more precise than on the scalp. Is that a question?
This is a late question. But
Yeah.
after you remove a part of the skull, how do you get it back on?
That's a good question. I've never done brain surgery. I mean, I think that I've been told that like
I mean, I've done it with rats and mice, and like not like that though. I mean, if you drill a hole, you can fill it with like a waxy.
yeah, they kind of have like a
foam
putty type
substance
stuff.
that
Yeah.
In
regrows.
the picture you just showed us, they're missing an entire segment.
Yeah.
My guess is they have that bone section, like they have actually taken it off.
Yeah. You take it off temporarily and save it, and then put it back.
And then they put it back on.
Yeah.
I mean, think of a baby, right? Their skull isn't completely formed. They probably put some new stuff back in there, and it will eventually. Think about breaking a bone too.
Yeah.
The bone will eventually rejoin. It's like a pretty
Yeah.
crazy, and that's why they're sitting in the hospital.
Right. Yeah, you don't do it lightly. But yeah, they have some kind of sealant or caulk that they use to kind of stick the removed portion
I'm
back
saying,
on.
like, wax.
I'm not sure what kind it is. Yeah.
Yeah, it actually is like bone, helps the bone grow back.
Yeah.
You can stick it in the hole when you do brain surgery. The skull back.
Yeah. And you can, I think they often have like a temporary plate that they will put in for some things, Yeah.
So what's the readout for EEG quickly?
Okay, yeah, so quickly, so we'll skip. So there's also stuff called MEG. It's very similar, but we don't need to get into it, really. Okay, so let's look at what so this is what your head looks like after you've had EEG. Yeah, you've got this gel stuff that you have to put on. So the nice thing about EEG is it's cheap and has good temporal resolution. So yeah, putting this in action here. So we did oh wow, it's already 3:30. Okay, so just to show you a quick study. So we did another study that's a very similar design. I'll just give you the quick upshot. So in this study, people either saw, so Sarah I think actually did this, saw either two faces, two words, or two pictures. Then they saw an arrow cueing them to think back to one of them, just like we saw in the previous study. There was also a condition where instead they saw a big green dot which cued them to press a button. or they saw a small green dot which cued them to just do nothing. And the idea was just basically to find out for this like active mental attention what the EEG signal looks like. That's the short version of this story. So just to show you what the signals look like, this is the signal you would get out of a single electrode. Well, each of these is. They have names. You don't have to worry about the names. FC is like here. PZ is like up here. And what you get is this is time on the x-axis in milliseconds. This is microvolts on the y-axis. And each condition is plotted in a different color. So for example, you can see in this case, the red condition is when they're pressing the button. So one not so interesting thing you can see is that about 600 or 700 milliseconds after the cue to press the button, you see this giant spike in activity in both of these electrodes. That's not that surprising because like anytime you move your hand or something, you get these giant EEG signals. So that was actually kind of just a control condition. We didn't care about that much. But if we look at other electrodes for when people are directing their attention to one of the things in their memory, so these were 2 electrodes where they showed a nice effect. So the blue is where they're thinking about something, and right now we're not worrying about what. It's either a face, a scene, or a word. And green is where they're really doing nothing in particular at all. What you see is you kind of get a nice little spike of activity early on. That's about 400 milliseconds. We're not so worried about this stuff, but don't worry about why. But about 400 milliseconds after you see the cue to think about the thing, you see this nice little initial spike. And then what's interesting is it then kind of goes below the baseline condition for a while and then rises up again. And you see that this is an electrode right here and another one kind of in the back of the head, which very roughly speaking, are kind of over or near to the areas that we were looking at in the previous fMRI study. So we think they might reflect similar underlying brain structures. And so our idea here about what might be happening is that this initial peak might correspond to the initial decision to direct attention to something, which might take about 400 milliseconds to kind of reach that decision. And then after that, at about 900 or 800 to 1400 milliseconds after you saw that cue is when you're actually kind of activating that area of your brain that represents that visual information, which is why you kind of see a little bit of sustained activity there for a few hundred milliseconds. So that's what we typically get out of these EEGs. And then we can look at other stuff, like we can look at the EEGs for different material types. The short answer here is that we found not much of a difference between whether you're thinking of a face, a scene, or a word. You can see there are small differences, but they're not statistically very reliable. And then there are crazy things that we can do, which I will not get into because they involve crazy amounts of statistics. I like this comic, haha. But the basic idea is that you can like take these very tiny differences and try to decode overall patterns of activity in the brain to see things that you can't necessarily see at a single electrode or time point. And what these graphs basically show is if we look at it that way, if we apply these really complicated pattern analyses, we can actually tell above chance, not super great, about 52 to 55% of the time, we can tell which of the 3 categories you're thinking of just from EEG activity. So that's kind of cool. You can kind of do some brain reading, blah, blah, blah. So that's probably about all we have time for. So there's other cool pattern analysis stuff we can do. I can't tell you about the study where I got to watch TV in the scanner for like 20 hours of my life that I ran this fall, this winter. But anyway, if you have other questions, let me know.
That, guys.